Good morning, everybody. Good morning from San Diego. This is the Neuroscience Gateway using Neuron and EEGLAB webinar. I'm Amit Majumdar from San Diego Supercomputer Center at University of California, San Diego. And I have my colleagues, Shubha Sivanyanam and Kenneth Yoshimoto, as well as many of other speakers and colleagues. So just a quick information about the agenda. You may have seen it already. I will do a very high level quick intro of NSG. Then Shubha will demo how to use NSG via portal and programmatic REST access. Ted Carnivale from Yale University will talk about Neuron and how to use Neuron using NSG. Hopefully we will have a short break. Then Arno from Schwartz Center for Computation and Neuroscience at UCSD will talk about EEGLAB and how to use EEGLAB, and then we will have a question answer session. As you know, the meeting is being recorded. We will make the recording available after a couple of weeks. Just so that you know, if you got a question, you just type it in the QA box. That's the only way you can communicate with us, since there are many of you, only few of us. And we will try to answer either over the QA text box or audio. And if you have questions in the future, please email nsghelp@sdsc.edu. So the Neuroscience Gateway is a primarily web-based science gateway that allows many neuroscience modeling and data processing tools to run on high performance and high throughput computing resources. It is free and open to any academic and non-profit researchers from any country pretty much. And given that it's free and open, and given that someone needs access to high performance computing for modeling and data processing, it allows everyone to use those resources for research and education. So as I said, the Neuroscience Gateway provides access through a web-based interface, the browser interface, or for more bit sophisticated users, programmatic access. And the main users of NSG are individual neuroscientists, graduate student, postdoc researchers, like all of you listening today. But there are some projects, community neuroscience projects, who can also send their computation to NSG. I will mention it later. So we have many supercomputers, academic supercomputers, that are available in the US and coordinated by a virtual organization called Exceed that provides the supercomputing power at the back end of NSG at various sites such as Comet and Expanse supercomputer at our center at SDSC at UCSD. But we also have machines at other centers at University of Texas, Indiana University, Pittsburgh Supercomputer Center. A new neuromorphic computing design at UCSD will become available to NSG also. In the year of 2019 and 2020, the US National Science Foundation has funded multiple new supercomputers at various sites. Over time, we hope NSG will have access to those machines also, including machines that are focused on AI research. So NSG provides various neuroscience modeling and data processing software. Initially, when we started in 2013, it was primarily computational neuroscience software, such as Neuron, Genesis, Brian. Over time, more and more cognitive neuroscientists and experimental neuroscientists have started using NSG. So you can see, Matlab, EEGLAB, FreeSurfer, and then more data processing like R, Python, and then even machine learning tools like TensorFlow were made available. Usually we listen to users and provide neuroscience software that are robust, have a good user base, and do need high performance computing. If some software is not used or not supported, we retire those. Just to show how NSG has grown over the last 7 or 8 years, if you look at the plot on the top right, you will see how much supercomputing time we have acquired each year starting 2013. You can see initially we were getting few 100,000 hours. Then over time, in the last few years, we are kind of settling at 12 to 13 million core hours on various supercomputers. And then if you look at the plot on the bottom left, you will see the number of users of NSG has also grown during that time, starting with about 100 users initially in 2013. Now we have about 1,200 users. So that's a good balance of requiring more computing time, having more users, and the number of publications and posters and presentations and MS thesis work using NSG has also grown. I think those numbers are not very accurate. My guess is the numbers are more than what's reported here, because we request you, when you use NSG, to please let us know your publications, as that is very important. Now, to give you a little bit background information about NSG, getting an account on NSG is very easy. You go to the web page and just fill in your institution, etc. information, and within a day or two, you will get an account. But the question is, how does the supercomputer time comes, given it's free and open to everyone? What happens is the NSG team writes a allocation proposal every year on behalf of the neuroscience community, that is all of you, to get time on these various supercomputers in the US. That allocation proposal goes through a rigorous peer review process, because many disciplines, from physics to engineering to social science to geoscience, everybody compete for the supercomputer time. But we have been successful to get time, and once we receive the time for a 12-month period, we try to manage the time for users like yourself in the best way possible. If someone needs more time, we work with him or her. But if one person started using a lot of time, we have to ask that person to slow down so we have enough time for all the users for the whole year. And then to get time for the next year in our allocation proposal, we have to show that we have pretty much used all the time. If we have unused time, that doesn't look good. So as number of users increase and more neuroscience software we make available, you let us know about your publications. All this information go in the next year's proposal. So another thing is, if a US-based researcher, say suddenly himself or herself needs 5 million core hours, we cannot provide that to that researcher, because that will be half of what we have for the whole community. We can work with that researcher, for the US-based researcher, to have her or him write their own allocation proposal. And once they get time for their own research, they can still use NSG. At the back end, they will be charging to their own allocation. Here I want to talk about two computers, supercomputers that NSG uses. You don't need to worry about all the numbers that I have here. You will see the point I'm trying to make. So Comet is the current supercomputer that NSG has access to, and it will retire in March 2021 as an NSF supercomputer. You see on the left some system configuration of Comet. But the main point I want to mention is each node on Comet has 24 cores and 128 gigabyte of memory. So per core, there is over 5 gigabyte of memory. But if you look at the Expanse supercomputer, which is just becoming available to NSG and to the US academic community, it will operate for 5 more years. It has 128 cores and 256 gigabyte of memory. So per core memory is almost less than 2 gigabyte. So the main message is, in the future, as Expanse becomes available to NSG, for many of the neuroscience software, you will have to specify how much memory you need, because multiple jobs, one could be the NSG neuroscience job, another could be a engineering users running a computational fluid dynamics job, will share the node. As long as you mention how much memory you need, our scheduler will schedule the jobs appropriately. So I mentioned that NSG is primarily used by individual users like all of you here, hopefully, but there are some big projects, neuroscience projects, just like the open source brain, which is well known among computational modeling neuroscientists. They send their large scale computing jobs to NSG. So a open source brain researcher may not even know that his or her job is running at San Diego Supercomputer Center. Open source brain is managed by researcher Parry Gleason and Professor Angel Silver in University College London. And we also work with underrepresented institutions. So here is one example. HSI STEM is a group of multiple HSI universities, and we do interaction and collaboration with them to see how they can use NSG for education, outreach, and training. We also have research experience for high school students. I will talk about it later. So just to give an idea of how NSG has been evolving, because the research and computing need of neuroscientists is also evolving. Initially, as I mentioned, in 2013, it was mostly computational neuroscience tools that were provided on high performance computing resources, that is kind of large supercomputers. But over time, more and more cognitive neuroscientists and experimental neuroscientists started using NSG, and they process lots of data, but in a single core or single node. Hence, high throughput computing is what needed for them, more than high performance computing needed for computational neuroscientists, as well as various large data transfer, data sharing, data access by multiple users. So these are the new features that NSG is starting to provide as more users from data science side join NSG. There are more and more training and workshops happening using NSG in recent years. And another thing we found from the community is that some of you are developing neuroscience software and you want NSG to be a dissemination platform for neuroscience software. So that's another angle NSG is taking in the coming years. So just to show you our pictures how NSG is evolving. On the left, you see the original NSG, which has both web-based and programmatic access. And you can see under compute resources on the left, under original NSG banner, it says HPC because it was for computational neuroscientists. Now, if you go to the evolving NSG picture, you will see that we are providing or trying to serve the experimental and cognitive neuroscientists who do a lot of data processing on using high throughput computing of open science grid, commercial crowd, as well as ability to transfer data. And then you see the tool developers and tool development environment, which means we are trying to make NSG a platform for tool dissemination also. And on the bottom, you see a EEG focus project. It's called NEMAR, Neuro Electromagnetic Data Archive and Tools Resource, funded by NIH, where primarily initially EEG data, bits formatted, which are uploaded to the Open Neuro project of Russ Poldrack, will be brought down to San Diego Supercomputer Center storage. And at the back end, it will connect with NSG for compute. But for easy researchers through the NEMAR web interface, they will be able to look at the data, search the data, visualize the data, do more kind of processing across data, meta-analysis, and all of this. I want to show some of the usage characteristics of NSG. So on the top left, there are a lot of numbers, but I will just explain. On the top left, you see the maximum core count job that has been run on NSG on various supercomputers in the last year. And you can see the trend line is saying, almost about 10,000 core jobs for computation neuroscientists. On the bottom right is the same plot, except from the beginning of NSG, like January 2013 till now. You can see a lot of large core count jobs were run primarily by computation neuroscientists. So this is for maximum core count job. Here I have the same plots on the top left and the right bottom, but except for minimum core count jobs. So you can see there are few core count jobs, 10 or so or 5. So this means over time, more and more data scientists, cognitive neuroscientists have been doing single core or few core jobs of processing their data. So this is good. This shows that more and more cognitive neuroscientists join over the last few years. And the plots before show that computation neuroscientists has been running bigger and bigger models. This shows the number of jobs that ran on NSG since January 2013. You can see some years, or some months, there were 70,000 jobs run, but typically few thousand jobs run a month. So to summarize, NSG is free and open. We provide neuroscience software for both modeling and high throughput computing for data processing. NSG is trying to be a software dissemination platform. We have many workshops and training, but the last two bullets are very important for us. If you use NSG for your work, please cite this paper in your presentation, publication, MS, PhD thesis work. And if you publish, present, or use it for MS, PhD, please send us email and let us know. These are very important for us to collect because we use that to get next year, millions of hours supercomputer time for all of you, and also to write funding proposal to keep NSG going. So we have many collaborators. I mentioned the one that are directly involved with NSG, Shubha, Kenneth, Marty, Steven, and me from UCSD, and Ted from Yale University, as well as we have many other collaborators, Scott McGee, Arno, who will speak, Ramon Martinez, and Young Trung, as well as Gart Cowenberg at UCSD. Here I show two undergraduate students who did summer internship in 2013. They are now, they were high school student then, now they're undergraduate student at Gonzaga and UCLA. And we have multiple grants from NIH, NIBIB, and NIMH, as well as from NSF on various projects that I already talked about. And we have the advisory board of Maxim Bazanev, Dieter Yeager, Stephanie Jones, Bill Litton, and Vinod Menon. Thank you. If you have questions, please put in QA. And I think Shubha will take over next. So I will stop sharing.
Are there any questions for Amit? If not, I'll start sharing my slides. Can you all see my slides?
Yes.
Okay, thanks. Good morning. My name is Subha Sivanyanam, and this session is about how to use the Neuroscience Gateway for running computational or data analysis codes on supercomputers. So during this talk, I will introduce the two ways by which you can access the Neuroscience Gateway, mention some guidelines for better use of this resource, and show a demo of how to use the NSG. So the first step, to, before you start accessing NSG is to get an account on NSG. And as Amit mentioned, you can do that by filling out the registration form that you can find on our webpage at nsgportal.org. And it's a simple registration form you can fill out and let us know, what kind of research you're doing, what software you would need, what software you're planning to use on the NSG. And once the registration is approved, we will create accounts to access the NSG through both the portal as well as the programmatic access. Just to note that the accounts are separate, meaning that if you get an access to use both the portal and programmatic access, and you're using them both, but if you change the password on one, the change will not be propagated to the other side. So the accounts are maintained separately. So once you have an account to use NSG, there are two ways to access. The first is through the portal, which provides a web browser-based interface that allows researchers to access and use the neuroscience-related software applications that we have already installed on various supercomputers. And majority of our users use the NSG through the web portal. However, there are those who may need to integrate, accessing NSG as part of their workflows. And for that, we provide programmatic access to NSG through the REST services. And depending upon, if you want to get an individual account with us or come in through the community account projects that Amit mentioned, we have two ways of accessing the programmatic access. One is called the direct, where we require individual researchers, or you may be using a software which has already integrated the access to NSG as part of the software, and you're downloading and using that. So you would have to come and get an account with us, and we call that as a direct access. Or you could come in through the web applications of community projects where you don't need an account with us, but the admin of the community project would have an account with us and will just direct the jobs from the web application directly to the NSG. on behalf of the community project. So those are the two ways of using the programmatic access. Now, regardless whether you come in through the portal or the programmatic access, I just want to share some general guidelines for using NSG. Um, though NSG is created such that the purpose is to hide all the complexities associated with using a supercomputer, uh, we do want our researchers, especially if your code or your model is CPU bound, or it is memory bound, and it is a little bit performance sensitive, we would want you to identify the HPC resources which is best suited for your code. And as Amit mentioned, the resources that we provide, they all have different architectures, right? So they have different number of cores per node, the memory is different, and all of this information is available through the resource user guides. For example, if there is a Comet user guide, Expanse, all of this is publicly available, and you can take a look at it to see what is best suited for your code. The second thing is the length of the time for which your job will run, and this is something that we want you to estimate. And this is the time that your code will take to run on a HPC cluster. Now, that we call as wall time. Now, it's better to overestimate this time a bit, because all NSG does is take all the parameters that you specify as a user, and then translate that into a Slurm script to be run on the HPC resource. So we want you to overestimate the time a bit, because we don't want your job to be terminated in case if your job runs a minute over the time that you think the job will run. So the wall time is very important, because otherwise your job may run for um, 10 hours, and then it may get killed if you have not provided the right time for it. The final point is we would like you to upload your input model as a zip file, and this is the only way NSG accepts your input file. And the reason behind that is when we started NSG, we based the input file format on how the model DB has, uploads it, allows their users to upload their input files. If you're not familiar with ModelDB, it's a repository of computational neuroscience models associated with publications. And the idea was, once you've used NSG and you have a publication, you may disseminate your code through ModelDB. And so we follow the same format. So you can have any number of files, subdirectories, or even just a single file. But what we want you to do is to create a single folder or directory and put all those files inside that and subdirectories, and compress that top level directory as a zip file and use that as the input file. And this is the probably one of the most commonly asked questions. We always have users who miss the single step. Either they would not create the directory, or the files are distributed. They may have additional files along with the single directory. So please follow this format, and this is the only format that will be accepted for running on NSG. So with that, I'll show a demo of how to use the NSG portal, and I'll be using an example code from ModelDB, and there is no particular reason for choosing this code model. I was just looking for a Python code, one of the latest models that has been deposited in the ModelDB. And I know that later in the day you would have a demo of Matlab and EEGLab and Neuron. So I just chose a pure Python code just to show this demo. And you can, if you already have an account and you want to try it out, you can also choose an example code from ModelDB. So with that, I will switch over to our website. This is the nsgportal.org, which is our website. From here, you can access the portal. And if you have an account, you would use the information here to log in. If this is the first time you're using NSG, once you log in, you would find absolutely no folders here. You can create a new folder. And once, if you create a new folder, it will appear on the left-hand side along, just like how my folders are displayed right now. And with each of the folder, you will have two subfolders. One is called the data, the other is called the task. Now, data is where you will upload your input model. And tasks are, all the processes that are associated with creating and submitting jobs to the supercomputers. So I've already created a folder here for the workshop, and I'm just going to show you the data part here. And I have uploaded that model DB zip file that I just mentioned. And uploading data is fairly simple. So if you click on the upload file, and uploading is just browse, select the file from your laptop, and then save it, and the file will get saved under the data. And you can upload how many ever input model files that you want. So once you have uploaded your data file, the next thing to do is to create and submit a job. And for that, you would have to go into the task. And in the task, there are some steps needed. One is you have to select the right input file. You have to select the software that you want to run. And you need to set any necessary parameters. And this could be associated with the software. It could be the parameters that you need to specify for running a job successfully on the HPC resource. So I'm going to go ahead and create a new task just to show you how to do that. And here I have one input file. That's the model DB zip file that I have uploaded previously. I'm going to select that one and select the data. And the software that I want to run is the Python. I just want to choose and run the Python. So as you can see, these are the list of software that we have installed on various resources. And some are on Comet, some are on Stampede, which is at Texas. There is one on Expanse that Amit just mentioned in his talk. So I'm going to choose the Python on Comet. And again, while I'm choosing Python, the Python is not just running just pure Python code. Sometimes there are software that is just written using Python, for example, NetBind, BMTK. And you can have access to all of those while you're choosing Python. And we also have some Python libraries that are installed. So once you choose the Python, there are some parameters that are predefined and available for the users to set. The first one is the maximum hours to run. And this is what I've mentioned about regarding the wall time. So how long will this job run? So reading through the model DB README file, I know that it does not take very long to run. So I'm going to just choose 30 minutes. And this line here, which says, do you require an RNIB model compilation, is very specific to neuron codes, and which if someone is using NetBind might need that. So we don't need that for this Python code. And if there are multiple Python versions, they'll be listed here. And I'm going to just choose this as it's the only available Python version. And what is my main input file name? And looking through the README of that model DB file, I know that it is called generate params.py. I don't have any subdirectories and I don't have any command line options, and these are optional, and you may have command line options, and if that is true, that you can enter them here. I'm just going to run on one node and one core for this particular job. But say if you're not sure how many nodes or cores are available, you can just click on that. We have the extra information here, and it tells you that this is going to run on Comet, and the maximum nodes available is 72, and the number of cores on Comet for each node is 24. So with that, I have chosen that. Now I can, by default, all files that are returned back from the Neuroscience Gateway will be called as output.tar.gc. For some reason, if you want to change that, you can change that here. I can just say ModelDB test file or ModelDB output file. And I want to save these parameters. And once I've saved, I can just call this is my test run. I can just call it test. And once I'm ready to submit it, I can save and run the task. So what happens now is that once I click on save and run the task, what's happening in the backend is the Neuroscience Gateway takes your input files, takes all the parameters that you just selected, including what software you want to run on, packages that up, moves that over to the resource, builds the job submission script, submits it, and waits for the job to complete and emails you when it completes. So at this point of time, you can actually log out, and if you don't have to wait here to see if your job is completed, because it will email you once your job is done. So once it's done, you can come and retrieve your output files. But for now, I just want to show you how, in case if there's any error or something, where you can look for those errors. And that's under these task messages. The task messages just list all the steps that I just mentioned. So you can see that the job has been staged to Comet, and all the input files are staged, and the job has been submitted to Comet. And this is the job ID on Comet. Now, again, we as an SG have no control over how or when your job will run on the resource, because all these are, these are national supercomputers. Other than NSG, there are many other disciplines that use supercomputers, and your queue wait time is largely dependent on, who's running or what code's running. So we have no control over that. But once if your job is completed, you will get a notification. So if you, I'll just check if the job is completed. No, the job hasn't started running here, but I did run a job earlier, and I can show you how that would look. So once your job is completed, you would get the output file. And here, I specified a small db.tar.gc. I can simply download it, and the download will have my output file. So other than this, what other features we have is the cloning feature, which means that when you press the clone, what it does is it copies the exact same setup that you had for the previous job, right, including the parameters, and it will clone it and create the same thing, where you can go and maybe if you're running a parameter sweep study, or you just want to change some command line option, you can do that, and you can clone and run multiple jobs. So that's the purpose of having a clone for in the Neuroscience Gateway. So this is the, as the whole purpose of Neuroscience Gateway is to make it easy for researchers to use the supercomputer to run your computational neuroscience codes on the supercomputers. And these are the steps that are involved in using the portal. So at this point, if there are any questions, please let me know, and or Amit or Cindy can read it out. If not, I'll proceed to showing a demo of how to use the programmatic access.
Shubha, please proceed.
Okay. So other than using the portal, as I mentioned, the other way of using the NSG is through the programmatic access with REST services. And for this purpose, I'm just going to show how an individual user will use it, and not the community projects through the umbrella accounts. So for the individual research users, you would have to have an additional registration step where you will obtain a key for your application ID. And the purpose of the programmatic access is to use the NSG through, with the comfort of your own laptop or, desktop. So you have to set some environment variables like in the URL, password, and key. And all of this information is in our user guide. So, I'm just showing you a demo of this, but if you're really interested in using it through the programmatic access, you can look at our user guide. And there are different ways to interact with the REST API. You can use curl commands. There are some PHP, Perl, Python, Java libraries if you want to incorporate that. So there are different ways, but for this demo, I'll just show you using simple curl commands, because that's the way majority of our users use it. So just to show you how the programmatic access will look for maybe submitting the same zip file. So this is just a simple line, and you can, as you can see, you have to specify the username, the password, the key, and the URL. And the tool here is the Python, which is again the same thing that I did using the portal. And the input file will be in a Python example .zip, and I'm running it on the number of nodes 1. And most of the functionalities of the portal is available through the RESTful services, where you can list your submitted job, list the results, you can download results, and everything you can do this programmatically. If you're not familiar how to construct this curl commands, we do have an NSG tool configuration helper where you can put all the information that you want, like the number of nodes, the cores, the wall time, and we will construct the curl command for you, which you can copy, paste, and use it. So I'll just show you that demo of that. So this is from our main web page. You can access the NSG programmatic interface through the NSGR, and that will open the programmatic access web page. So you log in with your credentials. And once you are logged in, you have an access to the application management. So under the application management is where you will create a new application. And when you create a new application, you just choose, for example, if you're going to be running a neuron code on visual cortex. So you can create some name which is descriptive of that. And your authentication type is direct if you're an individual user using it, and some website and maybe additional contact person who are in the same research as you are. And then once you create it, and key will be created for you. And that's the key that you would use. And I've already registered an application and I have a key, and I'll be using that for this demo. So now if you're looking for the documentation of how to use this, here are some quick start guides, a user guide that you can look at for seeing exactly how to set up the environmental variables, how to submit, how to list, how to poll to see if your job has completed. And if you want to know how to create your curl command, there is this tool configuration helper that I mentioned. And with this, I can just demonstrate and show you how to create the curl commands for the Python job that I just demonstrated through the portal. So I'm going to choose the Python. And for this, I'm going to do the same thing. I want to run for 30 minutes. I'm not running neuron code, and I want to run with the same Python version. And the file name is generate underscore params.py in my case. And I want to run on one node, one core, and I will select my input file, which is right here. And I just click on the view, and that will tell me how my curl command needs to look like. And it's also formatted for copy and paste, so you can really copy this and use that. So now I have already set up my environmental variables here. I hope you can see the screen. If not, let's increase the font a little bit. And now if I want to submit it, I have the same exact curl command here, and I'm just going to submit this, the exact thing that I've copy pasted from there. So if I submit this, what it's going to do is it's going to connect to the NSG server, move my input file, and all the parameters, and exactly do the same thing the portal did. But only thing is I'm just doing it at the comfort of my laptop. So it will take a second because the file size is a little big. So, but once it's transferred over, you'll have the messages like this, giving me an indication of that the job has been queued. There was no issue with submission of this job, and then what date it was submitted, and what is the most important thing is what is the job handle. Now the job handle is something that you could see in both the portal and as well as in the rest, and this is the only unique way we can identify your job. So in case if you have any issues with your job or if something happened, you have to send us this job handle, otherwise we would not be able to help you debug the issue. So now you can do exact same things as we did on the portal. You can list, you can see that once the job is submitted, you can download the results, everything you can do just using the curl services. So I'm going to see, okay, so I am just going to see what is going on with this job. So this URL is the same as, this is our NSGR URL. So it's the same thing, and I'm just using the environment variable there. And then I know that the job information from here. So I'm going to copy, paste it, and use the job handle. So what this would do is it just shows me the same information, and I'm just polling it to see at what stage it is at this job. Then I know that it has been submitted to comment again, and it's probably in the queue waiting to be run. So once this is completed, I can just download the results from here, and I will have it in my laptop. So this is how you can use the curl to submit jobs programmatically to NSG. So as I mentioned, if you have any issues running on NSG, you would have to send us the job handle, which is, it starts with the NGBW-job, and that's a unique identification for, for us to identify your job and help you with it. You, as Amit mentioned, that we get time on supercomputers and distribute it for our users. But however, if you, as an individual user or as a group, you want millions of hours and you want to run, still use NSG, but you want to write and get your own time, we do provide assistance for writing proposals for bigger allocation, but you can still use it via NSG to run your jobs. If you have any feature requests or software requests or any just general questions, the email address is nsghelp@sdsc.edu. Please email us and we can help you with your question. As Amit mentioned, if you use NSG resource, please cite us, and the citation information is also there on our website. And you can find this, and you can refer to our this publication. Thank you, and if you have any questions, please let me know.
There are a few questions which I answered already. I think nothing needs to be answered live. So maybe Ted can start and we can answer through the QA box.
Okay, sounds good.
Okay, I'm Ted Carnevale. I hope you all hear me and can see my slides.
Yes, we can.
I'm going to be talking briefly about Neuron and then work through a short example of how to use Neuron on NSG. And without further ado, let me launch right into the first slide. Many of you know about the Neuron simulation environment, but for those who don't, this is a software package which is used widely for modeling circuits and neurons models that are closely linked to experimentation, that involve biological complexities of anatomy and biophysics, and perhaps electrical and chemical signaling as well. And it is a project which is under open source development, directed by Michael Hines at Yale, has a GitHub page entry, actually several repositories at GitHub that are related to the project, and supported by NIH and the European Brain Project. Here are some convenient URLs for you. If you're interested, you can find out more about it at these locations. We provide courses at scientific meetings, as well as independent courses and workshops and webinars. And we're in the middle of a webinar now about Neuron. The Neuron user community, as of this month, consists of more than 2,000 subscribers to the Neuron forum and mailing list. There have been more than 2,300 publications that we know of that have reported work that involved using Neuron. And in terms of source code for Neuron models, you'll find over 740 of them at ModelDB, which is a searchable database of published models. ModelDB contains also over 800 other published models as well. Well, about Neuron itself, you build models either through a programmatic process using Hoke or Python programming language, or a combination of them, or with Neurons GUI. And you can combine all of these things if you like to take advantage of the strengths of all of them. It has tools also for analyzing models to verify that what's in the computer is close match to your conceptual model. And very importantly, since these are procedural programming languages, at least with Python, you can do introspection into neuron models, Pythonic introspection. We'll skip over the rest of this because you'd expect there to be some way of launching simulations and controlling simulation flow. You need to have ways to stimulate your models, you need to make user interfaces. The Neuron has a programmatic way of adding new functionality to its computational engine in the form of new ion channel specifications or synaptic mechanisms or means of chemical signaling and other things, including artificial spiking cells. It also has a rather powerful reaction diffusion module in Python, which can be used to deal with diffusion transport, accumulation of chemical signals, reactions among them. And most appropriate and pertinent to our present discussion, it has 3 different ways of parallelizing simulations, and we'll talk about those shortly. And these are nice links you can go to if you want to learn more about some of these things. So let's talk about what, again, it is that Neuron is useful for modeling. And the reason it's of interest is because the moment-to-moment processing of information in the nervous system involves propagation and interaction of electrical and chemical signals that are distributed in space and time. And oftentimes the equations that describe these processes are very complicated, so it's difficult to have a clear understanding of them or good intuition about what's going on. And so when you reach that circumstance, that's when you need empirically based modeling to help you with understand your conceptual models better. So to think a bit more clearly about this process of how it is that we develop understanding about the physical world. The physical world, of course, is extremely complex. And in some ways, in many ways, not yet completely understood. And all of our understanding about the physical world is obtained by a process of simplification and abstraction. And in any given circumstance, you're never thinking about any particular thing in the real world in all of its ramifications. You're always dealing with a subset of its properties, a subset of its behaviors, of its attributes, okay? And we're quite accustomed to doing this on a daily basis. So for example, if you're crossing the street and you see a large truck moving at you, you do not have to be a trauma surgeon or a physicist to understand that you better get out of the way or something bad is going to happen. You don't need to have great complexity in your conceptual models that you need for everyday situations. But in the course of scientific research, as we learn more and more about brain cells, brain circuits, the signals that they use, and how these things are regulated and interact, hypotheses that people are formulating about how it is that brain function emerges from these properties are becoming much more complex, to the point where, again, in order to plan experiments, in order to understand and interpret the results of experiments, it can be quite difficult. And those are the circumstances under which you need or might benefit from resorting to a computational model, okay? So if you are in that circumstance, you need to be able to make a computational model that's a very close match to your conceptual model, okay? And the utility of a simulation tool is directly related to how easy it is for you to establish and verify that match. So if you have a conceptual model that's very complex and you decide you're going to implement it in C++, you don't get much help from C++. If you're going to, if it's a model that's in a particular scientific domain and you use a domain-specific simulation environment like Neuron for neuroscience models, that's where you get benefit from the simulation environment, okay? All right, so an example, practical example. Here's a side view of a CA1 pyramidal neuron, and we can see lots of dendrites branched all over the place, a lot of anatomical complexity, and this cell also has a great deal of biophysical complexity, many different ion channels distributed over its surface, many different chemical signals going on inside it also. But if your experiment involves nothing more than, say, applying a patch clamp electrode to the cell body and injecting brief current pulses that are low in amplitude, so they're below the spike threshold, and then observing the resulting voltage transients, then it may be sufficient to have, as your conceptual model, this very simplified ball and stick. That is a lot of abstraction and simplification. But with such a model, it is possible to very closely replicate many experiments in which an electrode is attached to the cell body and current is injected and the resulting voltage changes are observed. Okay, what about if we really want to dig into this conceptual model? Well, it does have an analytical solution sometimes, depending on the properties that you assume for the membrane and depending on the whether or not the dendrite expands or gets smaller in diameter from the cell body as you move away from the cell body and what that relationship is. But even that analytical solution itself is quite complex, and it involves functions that many of us are no longer familiar with, like hyperbolic cosine, hyperbolic tangent. And for those circumstances, then it's useful to be able to resort to a computational model. And this little block of code in Python and this little block of code in Hoke, either of these, if executed in Neuron, would recreate this model, and you could then add additional attributes as you like to be able to have as close a match as you wished between what's happening in the computer and what's happening in what would happen as a consequence of your conceptual model. All right. Now, what kind of models can Neuron handle? Well, it can handle models that have a wide range of anatomical complexity, ranging from single compartments or even artificial spiking cells to highly detailed reconstructions of biological neurons, and believe me, these can have enormous complexity, to large-scale networks whose elements may involve any combination of artificial spiking cells and simplified or detailed neuron models. In terms of biophysical complexity, these models can involve anything that is described by kinetic schemes, differential equations, or events, including commonly observed things such as voltage and ligand-gated currents, synaptic transmission, important properties of biological cells and circuits, and also it can include representations of artificial spiking cells. Now let's get back to how we represent the properties of models of biological neurons. In neurons, there are basically two kinds of signals that we're interested in. One is electrical, one is chemical. In both cases, there's something that moves, and there's a driving force that makes it move, and there's a conservation law that allows us to write an equation that describes the relationship between what's moving and what's making it move. And the forms of these equations are identical for electrical and chemical signals. They're both described by exactly the same mathematical formalism. So we're going to stick with electrical signals for right now. And here's a cartoon of a small region of a cell, small enough that the transmembrane current, that is the current leaving the cell from the inside to the outside, is uniform over its surface. And then we have these adjacent regions of the cell from which current may enter our small region by flowing through these boundaries. And all these arrows indicate the positive directions for the transmembrane current and the axial currents. And if we were to write the conservation law for this small part of the cell, we would say that the transmembrane current, which is the term on the left-hand side, the sum of the transmembrane capacitive and ionic terms, the net transmembrane current is equal to the sum of all the axial currents arriving from adjacent regions. Okay? Well, what if we have a very compact cell? So it can be represented by a single compartment. So something like, say, a very small stellate neuron, whose membrane current is going to be nearly uniform over the whole surface. And say we're able to maybe attach a patch electrode to the cell that gives us access to the center of it, and we can inject currents through there and record the intracellular potential. Well, in that case, our entire model consists of a single equation, okay, where this is our transmembrane capacitive, and this is the sum of transmembrane ionic currents, and what's on the right-hand side is the injected current. There's no axial currents from adjacent regions because there are no adjacent regions. Fine, that's good and simple. But what if there is a cell with significant electrical extent? Then we have many regions. We have to chop the cell into many regions in order to make these regions small enough so that membrane potential is membrane current is uniform. And we have a different conservation equation for each of those compartments, okay? So that's an awful lot of compartments. That's a lot of subscripts. When biologists look at neurons, they don't see compartments. They see branches of cells, and they're not interested in knowing that today, when we're thinking about, say, electrical signals, when we can chop the cell into relatively coarse pieces, they don't want to keep track of the fact that, say, compartment number 6 is the cell body, because tomorrow they may be interested in chemical signals which need a much finer spatial resolution. And so they may have to chop the cell into many, many more compartments. So the cell body is now compartments 27, 28, and 29. And that would drive them nuts. So instead, Neuron uses the notion of sections, which are continuous length of unbranched cable. And so when you look at a cell and you see all these branches, then in your mind, you do an explosion of the cell into its parts. And each of these little parts would be a section in a neuron model. And each of these sections is described by the cable equation. And of course, we chop each section into as many pieces as we need to get good spatial resolution of electrical and or chemical signaling in it. And when we've done that, just from the standpoint of electrical signaling, we end up with something that has an equivalent circuit that looks like this. So where a section will be chopped into many different pieces, each piece is called a segment. And within each segment is the node, that is, in the middle of the segment is the point at which differential equations are being solved that govern the membrane potential of that segment. And this electrical circuit represents roughly the equivalent circuit of this section, where these resistors represent the axial resistivity of the cytoplasm to current flowing down the inside of the section along the length of the section. This is the membrane capacity. These are the membrane capacitances of each section, and the little boxes here contain all of the ion channel representations for the section. So this thing is going to have this is going to be represented by as many ordinary differential equations as you have segments, and also as many additional ordinary differential equations that you need to describe each of the ion channels. So a given cell could be represented by thousands and thousands of ordinary differential equations, okay? Of course, what we're dealing with here, stepping back from it, is we're dealing actually with a continuous system in space that we're discretizing in space to get rid of this partial differential term on the left. This is the cable equation. This second order partial derivative is the thing that we get rid of by spatial discretization. But we're still left with the differential equations that describe the time course of membrane potential and each of these gating variables for each of these ionic currents. and maybe more ionic currents if there's more ion channels present. Okay. So fortunately, neurons notation is much more compact than this. So you can simply use in a few statements of Python, you can create an axon model that has the Hodgkin-Huxley mechanism in it, and that takes care of the entire model specification. When we say axon is a section, we get the basic part of the cable equation. And when we say what ion channels are present, we get all the other terms in the Hodgkin-Huxley equation. Okay. So in more complicated models of more branched cells that have many different ion channels, we just end up with many more and more terms on the right-hand side of this, and we have many more compartments in our model. Ultimately, we have something that we can then run simulations with. In this case, we built a user interface to this Hodgkin-Huxley model axon that allows us to see membrane potential as a function of distance along its length as the spike propagates from the left to the right-hand side, as well as the time course of the membrane potential in this model at a particular point. Okay, so again, how we build these models, we can do this with programmatic specification in Hoke or Python or through use of graphical tools. The graphical tools that are available are very powerful and very flexible. The import 3D tool allows you to import detailed morphometric data, say from Neurolucida, Eutectic, or the other SWC specification, which is commonly used. The cell builder allows you to create stylized models, ball and stick-like things, or artificially branched model cells, or to deal with managing detailed morphometric reconstructions. You can add other ion channels to this detailed model cell and create cell classes for use in network models. The channel builder allows you to create model specifications of ion channels using either Hodgkin-Huxley style ODE specification or Markov model transitions and with rates that are voltage or ligand dependent. And there's another tool, for example, the model view tool, which is very useful for analyzing models and presents you with a browsable outline that shows, with a few mouse clicks, reveals very quickly what is in the model and where things are located. For example, where ion channels are distributed and how they may vary with anatomical position in the cell. Very helpful, since again, you don't want to look at model source code and try to pretend that you're a CPU parsing that when you're trying to understand somebody's procedural code. Okay, so here we have on the left Hoke statements, and on the right Python statements that would recreate that ball and stick model that we were looking at earlier. Either of these programs fed to Neuron would generate exact computational model that matches closely your conceptual model, that ball and stick. Now, how is it that either Hoke or Python can be used? Well, because really at the core of Neuron is a computational engine that has data structures and compiled routines that are specialized for dealing with the kinds of problems that arise in modeling cells and networks of cells. And the Hoke interpreter and Python interpreter allow you to write statements that are compact and human readable that access these data structures and their parameters and variables to configure whatever it is you need to simulate. And also anything you can do in Hoke, you can do with Python and vice versa. And if Python needs to access some particular Hoke syntax, it can do that. And if you're operating in Hoke and you need to access some particular Python feature, you can do that. Of course, the availability of Python means you can also bring in any of the literally thousands and thousands of Python modules that are available around the world. Whether you want to do that or if it's safe, you have to decide. And if you need to add additional data structures or compiled code to Neuron's main computational engine, you can do that with a programming language called NModel. And I'm not going to spend any time talking about NModel, but NModel is capable of specifying very detailed things in a very humanly readable way. For example, this representation of calcium-triggered transmitter release, which involves many states and transitions between them, including buffering of calcium, radial diffusion of calcium, binding of calcium to vesicles, release of acetylcholine into extracellular space, and breakdown of that acetylcholine afterwards. And it all executes at machine language speeds because it is translated to C and compiled into a linked library that Neuron reads when Neuron starts up. Okay. Neuron has several different integration methods that it uses. Its default is a backward Euler, fully implicit Euler method, which is extremely stable. First order precision, of course. But then when you realize that the parameters that we know about biological systems are really known only to a rather limited degree of precision, and furthermore, there's tremendous variation from nerve cell to nerve cell of the same kind, even within the same individual, let alone variation between individuals of a given species. First order is probably adequate for most cases. Any phenomena, any biological phenomena that rely critically on the value of some parameter probably are too fragile to be robust for that organism. You certainly don't want vital biological functions to depend on critical parameter values. Think how much variation there is in your mental acuity throughout the day or after a long night of being up and being fatigued, for example. Neuron also has adaptive integration that is very easy to use. I'm going to skip over that because if you want to know about that, you can learn about that elsewhere. Neuron's utility for network modeling is particularly strong, largely because of its ability to deal with spike-triggered synaptic transmission. One of the simplifications, one of the conceptual simplifications that people make in implementing network models is to assume that transmitter release happens when the spike reaches, propagating from the cell's spike trigger zone, propagates down the axon and reaches the presynaptic terminal, at which point magic happens and transmitter is released that diffuses across the synaptic cleft to interact with receptors on the postsynaptic cell. Now, there's no magic actually, it's just the spread of chemical signals. But this can be represented extremely efficiently. Instead of having to solve the cable equation that propagates the spike all the way from the spike trigger zone all the way down to the synaptic terminal, instead it is possible to just put a spike detector at the spike trigger zone, and when a spike has occurred, then generate an event that is going to be delivered to the synaptic mechanism after a delay that is equal to the sum of conduction latency and synaptic latency, which is about 1 millisecond, okay? And that allows extremely efficient simulation of networks because we throw away all the equations that are needed, say, we don't have to solve the cable equation at all those little compartments between the presynaptic terminal and the cell body. We also get a lot of efficiency because suppose there's a presynaptic cell that projects to many synaptic targets. If you attach many of these things called netcons, which implement the spike detector with the delay and the event delivery, each of these netcons will share a common spike detector. You don't have to monitor that same point many times to see if there's a spike there. There will be only one spike detector, and each of the netcons can have its own delay and its own weight, which is the strength of the interaction, that is how much it perturbs the synaptic mechanism, okay? Also, this strategy that's used in Neuron gives us very efficient convergence. For example, suppose we have a lot of synapses of a particular type attached to a particular region of the cell, for example, basket cell synapses attached to the cell body. There can be 50 or more of those attached to a cell body. Each of them is GABA-ergic, and each of them, when it opens a channel, is going to be opening a channel that has a very similar time course, is going to have an identical reversal potential, the reversal potential of chloride, of course, to any of the others. So instead of representing those 50 different GABAergic inputs to the cell body with 50 different differential equations, simply have one differential equation which replicates the dynamics of a single synapse, and drive it with events converging from many different presynaptic spike sources, each with its own weight, and linear summation of synaptic conductances give you the temporal summation of synaptic conductance that you would expect. Okay, so this is extremely efficient because instead of having to deal with 50 different ODEs, just one ODE perturbed by different afferent spike streams. So to see how this works in practice, let's imagine a synapse with a fast rise in monoexponential decay. This is the key to it right down here, is this block which says when an event is delivered, what happens is synaptic conductance is perturbed by an amount equal to the weight of that connection, after which the synaptic conductance continues its monoexponential decay, which is specified by this derivative block. Okay, much more complex mechanisms are implemented and have been implemented for event-driven synapses, and they work extremely well. And in fact, this ability to deal with events has allowed neuron models to also take advantage of event-driven artificial spiking cells, such as this integrate and fire artificial spiking cell, in which every time an event arrives, the excitation variable, the membrane state variable M, is perturbed by an amount equal to the weight of that synapse, so that if you have two afferent streams, one with a weak weight, one with a strong weight, arrival of an event on the weak stream makes the membrane state jump by a little bit. Arrival of an event on the strong stream makes it jump by a lot. And if it jumps over one, this artificial spiking cell generates an output event and is reset its membrane state to 0, where it stays until a new event arrives. This cell takes almost no computational effort because it doesn't actually involve numerical integration. It only needs to be updated every time a new event is delivered. And after that event is delivered, you don't have to do anything more to it because you know what its future time course is from the analytical solution of the first order differential equation that describes the time course of M. So cells with extremely simple descriptions, whose future behavior is knowable based on an initial condition problem that has an analytical solution, can all be handled as artificial spiking cells for event-driven simulation. And event-driven simulation cells like that can be mixed in networks with biophysical model cells, and they can generate events that are delivered to each other. So you can have quite elaborate constructions of network models. As I said earlier, Neuron allows 3 different kinds of parallel simulation. One is bulletin board style, where tasks are posted to a bulletin board, and the workers pull these tasks off the bulletin board, grind on them for a while. Each host that does this grinds on it for a while, sends its response back to the bulletin board, and the master then pulls these responses back from the bulletin board. Requires very little effort to program that. This kind of embarrassingly parallel simulation is very common for optimization and parameter space exploration. Neuron also can do multi-threaded parallel simulations. This doesn't really require any programming effort usually. And more pertinent to our discussion is its ability to deal with network models that are distributed over multiple processors, in which the spike source and target may actually be on different processors. Furthermore, These models may involve very large, complicated model cells that may be split into pieces and distributed over multiple processors to get load balance. And it is possible to use these features to be able to simulate quite large networks. And I will skip over how this works, and if we have time, we can come back to that. But the point is that there have been many published models that have used this feature, and benchmarks of performance have been performed with these various models, and generally they scale quite well. So that if you increase the number of processors for such a distributed model, you get an appropriate reduction in runtime that you would expect if you had strong scaling, until you get to the point where each processor has so small a piece of the original model that it doesn't have much to do. If it runs out of stuff to do between, in the course of a simulation between when it's calculating and when it's exchanging data with other processors, then you have some wasted time and you need to maybe do a little more sophisticated load balancing, which is possible with Neuron. It is assumed that before you start doing this stuff on a resource like Neuroscience Gateway, that you will have worked through those parallelization issues, such as load balance, model setup time on your own hardware on a toy or downscaled model. And it's very possible to implement models that you can, by changing a single parameter, change the scaling of. And so you can do all that debugging locally. And then when you need to use it on the production simulations, you can move your code to something like Neuroscience Gateway and run the code there. Okay. At this point, I imagine there's a gazillion questions. Maybe, maybe not. Let me see here.
No, Ted, there is nothing I think you need to answer right away.
I think you're right, Amit. Very good. So let me then, let me go to Neuron on the Neuroscience Gateway. I'm going to share that. Okay, do you see this now? Neuroscience on the Neuroscience Gateway portal, Neuron on the Neuroscience Gateway portal. You see that
Yes.
slide? I hope.
Yes. Yes, we see that.
Okay, good. All right. So fortunately, it exists now. 9 years ago, it didn't exist. And I'll tell you, we were having a difficult time with it because Neuron's ability to deal with distributed models had existed for about 6 or 7 years. And the problem was that very few neuroscientists had access to parallel hardware that was more than, say, whatever was on your desktop, or if you were fortunate and you had a geek who could put together a small Beowulf cluster or something for you. So what we were doing oftentimes was helping people who had begged, borrowed, or stolen time from their local engineering departments or physics departments, where they might have had a few cycles left over after they finished simulating the universe. And you buy those people enough beer and maybe they'll give you an account and maybe you can play with your silly neural simulation tools. But helping people get set up on all these different platforms with all the different local policies that they used with regard to file management, queue management, et cetera, was a big pain. So it was a real relief to us when the Neuroscience Gateway was originated, because that meant that people could run their code on Neuroscience Gateway, where our programs had been installed and properly configured optimally, and where they had a nice uniform interface that Suba showed you. So who's actually been doing what? Well, we know about the 31 publications in refereed journals that we've been able to gather from users of Neuroscience Gateway. Again, we have to ask people, please tell us what you're doing when you publish. But we know there's also many, many posters and presentations at meetings that we've seen ourselves, and fortunately people are reporting those to us. And when we look at the topics that people are using the Neuroscience Gateway with Neuron to study, it includes things that range from optimization of large models of cells and brain circuits, studying mechanisms of generation of phenomena such as local field potential brain rooms and EEG, to study how it is that normal and abnormal brain function emerges from the anatomical and biophysical properties of cells, such as olfaction, learning, and memory, and epilepsy. To develop and deploy research technology, such as new methods for spike sorting, very important for people who are doing large-scale recordings from cells in behaving animals. Development of simulation software such as NetPine, which actually uses Neuron as its computational engine. Development of environments for model optimization and collaborative research. And also people are using this Neuroscience Gateway for simulations involved in development of therapeutic technologies such as stimulation of peripheral nerve or central nervous system, for, say, for example, deep brain stimulation, motor prostheses, sensory prostheses, neuromodulation. And there's a large amount of application in education, especially neuroscience education, bioengineering, and computer science. But one thing in common for all of these is before you start to use the Neuroscience Gateway, it's a really good idea to make sure that your code is debugged and you run some preliminary benchmarks so you have an idea of how much time you need, how many processors you're going to use, because it's a long turnaround. So if you have a problem that crops up when you're on the Neuroscience Gateway, you've thrown away time that you were allocated, and it's not a very tight debugging loop, because there is a turnaround time, much faster on your local hardware. Okay, so today for my example, I'm going to show you a Neuron implemented simulation of a cortical column involved in actually thalamocortical network. And this original paper was published by Roger Traub and his collaborators back in 2005. And they implemented their code. Actually, Roger implemented their code in Fortran on a very particular architecture machine. But it's a useful model, so it's been translated into to Neuron, not just in the original Fortran that it was. This involves many different types of model cells. The toy implementation of this that we're going to look at today has 356 cells, and the full model is 3,560. And as I said, the original implementation was very lab specific. It was designed to run on exactly 14 processes on a computer that only Roger had access to out of the whole planet. Tom Morse and Michael Hines worked with Roger to port that to G77 Fortran, and then to implement, Tom and Michael worked to implement a neuron model with that, which would work with any number of processes on any machine that has Neuron installed. Okay, now the translation is not a perfect translation because of the way that Roger implemented things such as branching, and he did many idiosyncratic things which don't translate well to anybody else's simulation tool, but you can make sort of a first order approximation to it. And if you want to know the details of that, definitely look at this model EB entry and read the README files. I downloaded the Neuron implementation, unzipped it, compiled the mod files, and checked the runtimes on my own desktop machine just to make sure that I wasn't going to be unleashing the Sorcerer's Apprentice. turned out that runtimes were quite reasonable, setup and runtimes were quite reasonable, so I didn't have any qualms about doing this with Neuroscience Gateway. So this is what I'm going to try to show you a bit about today. Suba pretty much took you through the steps, and we're going to basically just recapitulate those. I created a folder, I uploaded data to that folder. I think I might skip that because I don't want to stress my poor quality internet connection. And then configure a task, run it, check status, and download and examine results. So let me stop this particular share and start a new share. Okay. Good. Let me make sure that of what we're looking at here. You should be looking into my account at the Neuroscience Gateway. Do you see that? It says Traub 2005 up here in the upper left.
Yes, Ted.
Everybody
we
sees
see
that?
Yeah, Ted, we
Very
see
good. Okay, good. So what I've already done is I've already created a folder. And as Suba pointed out earlier, when you do that, whoops, I'm bounced. I'm going to have to log in. Log in. Okay, good. I've already created a folder. And as Suba pointed out, you're going to have within that a subdirectory for data. And I have uploaded the zip file for Roger's model. I got that zip file straight from ModelDB. I'm not going to bore you with showing you that. I just expanded my Firefox window. Do you see the expansion now?
Yes, Ted, we saw the expansion.
Did you see the whole Firefox window? Good.
Yes.
Good. All right. So you can see here already I have this data object which I uploaded. And when I first went to tasks, there were no tasks. So my first thing I did was to create a task. And I'm going to create a new task. And I'm going to call this Traub trial run. Okay. And my input data will be from my data folder of Traub 2005. And I'm going to click right there in the checkbox. I hope your screens have all updated now. And you see the checkbox is selected. And I will now click on select data. And it says one input set. Very good. So that's the source code that we're going to use. And it was a zip file. And I know exactly the information that I need from that zip file in order to be able to specify how the simulation will run. What tool am I going to use? I'm going to use Neuron 7.7 on Comet. Okay. And if I want to learn more about it, I could click on this eye, and that would pop up a window that would tell me more about this implementation. But I'm just going to click on Neuron 7.7 on Comet. And now it tells me, it reflects my selection. That's good. Now, what are the input parameters? The input parameters are what I would call my simulation and parallelization parameters. What are they? Well, as Suba reviewed, how long do you want this to run? Well, I'm going to leave that at a half an hour. It won't take anywhere near a half an hour. I'm just going to leave it at half an hour. That's safe. The main input file name is, in fact, the file name that has to be executed by the simulator in order to launch the simulation. This is the correct choice for me. This default of init.hoc is the correct choice. If I needed to type something else in, I could do that. Do I need to run Neuron with a single extracellular layer? No, I don't need to do that. I'm not using extracellular, and I don't need to use single extracellular layer. It is in Hoke. I don't have to say it's in Python. There's no subdirectories I need to specify. The command line option, I need to tell my program, my program init.hoke is looking for a command line option that tells us when to stop. And so that is a command. So it's -C is the switch. And then I do single quote, my T stop equals 100. And there's my closing single quote. And then I hit enter. Then I come down here and let me go back. Did that get set? Yes, good. Okay. So I see that my runtime that I specified is indeed 100. Very good. Fine. How many nodes? I know there's what, 32 nodes on, excuse me, no, I know there's a bunch of nodes. I don't need any, I don't need more than one right now because I'm only going to be using a handful of cores. And I'll just let them all run on one node because I think that'll be adequate. Okay. In this case, if I wanted to, I could say use just one core and just see how slow the simulation could be. But I'll leave it at 8. A default of 8 is fine. I just want to see that I get a result. Okay. And the name for the compressed output file, I'm going to call this test run. So there will be a zip file, test run result. Okay. That's a long name, highly mnemonic. If I was doing something very systematic, I might instead include a hash code in that name just to keep track of all the parameters that I set properly and make it unique. But that's adequate for now. I don't need any advanced parameters. I will click on the Save Parameters button. Save Parameters. Now let me just look and verify that the parameters are what I want. Click on the 8 parameter set. Because even though this takes a little bit of time to check these, it's going to take a long time to run, a much longer time to run the simulation by comparison to the time I used to set this up. And if I've made a mistake, I'd better catch it now. Okay, so let me go back. This is fine. Let me just click on tasks. And where is, whoa, I didn't save the task. Oh dear. Well, there's a mistake. But wait a minute, continue editing current tasks. Will that save me? Trial run. I didn't mess up too badly, did I? So let me just save the task before I destroy it. There we go. Trial run. Okay, let's just look at those parameters one more time. I'm just checking it, and I'm not going to show you it worked. I didn't destroy anything. Okay, so there I am. There it is. So this, if I want to run this, I click on Run Task, and it gives me the warning here, etc. Okay, fine. I will be patient. Click on OK. And now we're off to the races. Okay. It says View Status. I can click on View Status. And I see these task messages, and it says it's in input staging. That's very nice. I'm being told that my internet connection is unstable. So before, you probably got some garbled voice there because my internet connection does appear to be unstable. This will eventually return. Let me go back to the task list, and I'll show you one of the results. Okay. So here's one that I ran that ran on two machines, so that I could actually get a total of 32 processors and not cut into the memory that they were sharing. And here is my output file. And if I want, I could examine that in detail. And let me see here. Can I view this? Ta-da, how do I view this? Can I look at this? Click on an output file below to review its contents. Well, let's see. View current task. I don't know if that's going to show me anything. That's not the contents. But anyway, what I would do is I just download the darn thing to my own PC, expand it, and get results. And if I want to see those, let me see. Let me just stop sharing, and I can show you now on my own machine the results. Okay, share. And this is on my own machine, CD results, CD N2C32. And here is the major result, Traub, out 32 dat. Okay. And this shows me all the different times at which cells fired and the identity of the cell that fired. Okay. And I can dig into these and analyze and plot results and see if I got something that's useful to me. So that's about all I have to say. Are there any questions? Okay, if there's no questions, it looks like I'm ready to pass the baton. And at this point, I'm actually going to have to leave the webinar because something has come up in the interim, and I'm just going to have to quit now. I will be available by email afterwards, and I imagine you will see many useful things in the remainder of the show. Okay, thank you very much for your time.
Okay, thank you, Ted. Just to maintain the time, instead of 15-minute break, we will take just a 5-minute break, and we will start at 11:15 the San Diego time. So just 5 minutes break. Thank you.
EEGLAB and how to use EEGLAB on NSG. And so first I'm going to introduce EEGLAB and show you some basic processing using EEGLAB. So the first part doesn't have anything to do with NSG. And in the second part, I'll show you how to use EEGLAB on NSG, and I'll try to do some demo. So we'll have to cross our fingers that everything works fine. And so first, what is EEGLAB? Well, EEGLAB is about interpretation of scalp EEG signals. So when you record brain waves, you record signal from individual electrodes. And the work is to actually try to interpret these signals and understand what it means in terms of what's happening in the brain. So that's why we need software to process this type of data. And this is the outline of EEGLAB. So it's been around for about 23 years. First, it started as the EEG ICA toolbox at the Salk Institute. And then we had the first version of EEGLAB it was just doing artifact rejection. It was also at the Salk Institute, and where both Scott McCaig and I were working at the time. And in 2003, we issued a release to a wider audience. So we had a massive mailing list, and we started getting NIH support shortly after that. In 2006, we implemented a plugin mechanism so people could plug in their own code. And we believe also that's one of the reasons EEGLAB is so successful, is that we have right now 130 plugins, and we have a plugin manager where people are in EEGLAB and they can download these additional extension to do custom processing. And I'll show you how to use that, because we'll use the NSG plugin for EEGLab. So I'll show you, we'll download it and install it. And in 2011, there was a paper showing that EEGLab was the most widely used EEG research environment among commercial and academic softwares. In 2018, we had a drastic change in EEGLab. So EEGLab only relied at that time on, before that, it relied on computing averages at the group level. And in 2018, we changed it, so everything was done at the single trial level. So of course, much more disk and memory requirements, but eventually better statistics. And we also paired with Cyril Pernet at the University of Edinburgh to do general linear model on EEG in the same way that you would do that on fMRI. And I'll talk about that a little bit afterwards. And in 2020, and that's tightly linked to NSG as well. We're in the process of finishing BIDS integration. So BIDS is the brain imaging data structure. It's a type of file format for neuroimaging data, and originally was for MRI, and now it's also for EEG, and creating big data pipelines. So I'll talk a bit about that. And the big data pipelines are running on NSG. And so in a few numbers, EEGLab, it's about 600 functions, and they're all standalone functions. So each have their own documentation. And it's about 70,000 lines of code. It runs in a graphic interface, but actually as you use the graphic interface, it writes code for you that you can later reuse. There's about 400,000 downloads over the past 10 years. We have 15,500 users on the diffusion list, which is a list where we send new announcements, and we have about 5,500 on the discussion list, where there are messages every day, people discussing EEG processing. We did a research in 2017 about the paper that were supported by the National Institute of Health and citing the used EEGLab. And we found, when we total all these grants, NIH grants that were using EEGLab, that it was about supporting $288 million of research just in the US and just for NIH and NSF. We've had NIH funding since 2003 to support the development and continued development of EEGLab. Here it says 75 plugins. That's an old slide. Now there's 130 plugins, so I didn't change that. Also, every time you start EEGLab, unless you uncheck an option, it's going to ping to a server that's called mixed panel. So we know that approximately 140,000 EEGLab sessions are started every month from about 10,000 unique users. And the number of sessions varies widely. Some users start 500 sessions, some users just start one. But that's to give you rough estimates of how many people use it in the real world. And this tool also allows us to see which version people are using and other kinds of details. And this is where it's spread out in the world. So strong concentration in the US, Europe, and Asia, mostly. So this is other statistics. So this is in 2011, the paper that showed that EEGLab was the most popular software. There was a survey among people doing neuroimaging and EEG research. And you can see, so EEGLab was the most used, at least twice more than field trip. And also EEGLab or field trips are tightly linked. You can actually go back and forth between EEGLab and field trips. So, and then you have Brainstorm, Brain Visualizer, which is the first commercial software, etc. There has not been another survey since then, but we monitored a number of citation to our article through Google Scholar. And then we can see that it's still increasing linearly with about 4 citations per day. So people are still continuing to increase in using EEGLab. And you might say, well, now there are tools, so what's the advantage of using EEGLab? So we also compared to other tools, in terms of what you can do and the use of doing different things. For example, EEGLAB versus field trip versus brainstorm, etc. Whether they're open source or COM means commercial, and type of user base, whether it's Matlab based or not Matlab based, whether you can do time frequency decomposition, source localization capabilities, plugins, statistics, ICA, group level analysis, etc. And cloud and GPU computing, again, that's an old slide here. I should say yes, because now we have NSG, which I'm going to demo today. You might also wonder, well, I mean, now there's Python. Why should I buy Matlab to run my neuroimaging processing on EEG? Why not just use Python? And on Python, there's, for example, the MNE software. So the MNE software was developed for MEG, but you can also do some EEG processing. Here, these are the statistics of citations as of the end of 2019. And you can see MNE, which is Python. All the other ones are Brainstorm, Fieldtrip, EEGLAB. All the other ones are Matlab. It's less than 10% of the citations. So people still mostly use Matlab. And that's actually a stronghold of the company that does Matlab, the MathWorks. So they cater to us and they support us because in neuroimaging, that's one of the fields in which Matlab is still widely used. And so that matters because, about the user base, well, if only 10% people are using MNE and Python, the type of support you'll get and the type of robustness of the function you use will be much lower. And we also have a page using EEGLAB versus Matlab versus Python. Even though Matlab is commercial, it's very cheap when you're a student, and usually your university has it, so you don't even have to pay. Also, one argument is that Python is like a step up in terms of programming language. So a step up in the sense of complexity. Python is much more for programmers. So you need to spend much more time learning to program when you use Python. When you use Matlab, Matlab is much more intuitive. You can just type a command, plot things. Matlab and R are at the same level, and Python is a little bit more abstract in terms of programming language. So you have to spend more time learning the language. There's all the Python libraries, which are headaches, etc. So you don't have that problem with Matlab. So if you just want to get the job done, there's strong arguments for using Matlab-based tools. And again, there's a web page where we compare, the advantage of using Python versus Matlab. And you can actually use EEGLAB on Python as well, because there's a library that's actually very robust. That's called arc2py, and that translates that allows to execute any Matlab code on Python using the Octave engine. And so there's also examples. So even if you're using Python and there's some functions in EEGLAB I wish I could use, you can do that. So I'm going to show you a processing pipeline, which is our standard processing pipeline, and I'm not going to go into detail here. It's going to be very high level. So if you don't know anything about EEG processing, you might be lost a little bit. So this assumes that you know something about EEG processing. So the first thing, so we have EEG raw data. So we're going to import the data into EEGLAB. We're going to remove unwanted channels, and then we're going to use one of the plugin of EEGLAB that comes by default. It's called the Clean Raw Data plugin. It just cleans your data very well. And I'll explain a little bit how it cleans the data. Then we're going to change the reference of the data to average reference. We're going to run independent component analysis, which is a method that our lab pioneered. And in this case, you can run independent component analysis, or ICA, to look at sources, but here we just reject artifactual components using another plugin that's called IC Label. And this is also a default plugin of EEGLAB. So this is the plugin to reject bad data. And how does it reject bad data? Well, it looks at correlation between channels. So here you see these matrices of 64 channels, and I do the pairwise correlation. And you can see this is good data. So the channels are highly correlated, and this is bad data in the sense that some channels here are not correlated with others. So these are the channels that would be removed from the data. So we set a threshold on the correlation, as I'll show you, to reject these bad channels. And then there's other process in which we try to find, so these are to detect the bad channels, then we have to find the bad portions of data. And to detect the bad portions of data, we use artifacts of space reconstruction, except we only use the part of the algorithm that detects the bad portions of data. And this is a complex algorithm that's based on projecting the data on principal components and try to see if there's outliers on these principal components. And if there's outliers, it usually tries to correct for these outliers by reconstructing the data. In this case, we'll just reject the bad portion of data because we don't need to keep the data corrected. And then I mentioned IC labels, so independent component analysis. We have lectures online about independent component analysis. So I assume you've heard about it, but basically on raw EEG data, you can run independent component analysis, and then it's going to find independent contributor to the EEG data. And these independent contributors, they have scalp topographies, they have time forces like EEG channels, and they often represent brain activity or artifacts. And so IC labels, so Luca Piontoninashi was a PhD student in our lab, and he designed that tool in which first he crowdsources labeling of ICA components. So he put 15,000, he put 100,000 components on the web, and then he had a contest where people would label components and compare their performance with experts. So here you see this is when you label a component, so you say, oh, do you think this is brain? Do you think this is muscle? Do you think this is eye, heart, etc.? And then you can see what other experts are thinking about that. So you can not only label them, but you can learn. So there's a section where you learn about recognizing these components, and there's a section where you label these components. And so you have this contest. You could win an iPad. Sorry, it's not over, so you can't join the contest anymore, about who would label the most components. So you had a total of 15,000 components labeled by experts. So then you would know, okay, this component is an artifact and it's an eye artifact. This component is a brain component, and this component is a muscle component. And again, it takes expertise to recognize these. So this is why he had this contest. And once he had all these labels, he trained a machine learning model to automatically detect these categories. So these are the categories. And in his paper right here, he also compared with other methods. There's actually other plugins in EEGLab to label components. So that's not just the only one. That's the one I'm going to use here. But if you talk to other researchers, lots of them use Mara or Faster or Adjust, which are other automated component labeling techniques. And you can find all these plugins in EEGLab. He compared them and found that his method was better than the other ones. So I'm going to use some data also when I'm going to use the demo on NSG. And I'm going to use some data from OpenNeuro. So OpenNeuro, it's a platform. It was originally designed in 2016, mostly for fMRI and MRI data. And now it's available, and now it's been open to the EEG community. We also got a grant from NIH to develop a platform specifically for EEG based on OpenNeuro. It's called NEMAR. I'm going to talk about this a little bit later, but here's just to show you what type of data I'm going to process as a demo. And this is the data which is we commonly call Hansen-Wakeman, because these are the two main authors of the paper. This is a face experiment, face categorization experiment. And so you can download the dataset at this address right here. So that's the dataset I'm going to use. And this is a dataset. So they released the data, and they released the data on the Open Neuro platform as well. And this was EEG, MEG, fMRI, MRI. So there's lots of data in there. And so we stripped it down. So we only have the EEG, and we also resampled the data. So I think the original data set was 48 gigabytes. So we stripped it down to 4 gigabytes just for EEG tutorial processing. So that's the data set we will use. The data set is also on Open Neuro. It contains all the detail of how we derived that data from the original data set. And it's a data set that's in bits format, so it's a brain imaging data structure. And so this is the data. So here we have 3 types of stimulus: famous faces, so you see Brad Pitt; non-famous faces, the unfamiliar face you see here; and then scrambled images. So that's the third image. And we have 3 types of repetition here, although we're not going to look into that. So they're presented first time, second time, and then a third time. And so this is how the subject look like after you download them. This is just part of the folder, but basically you just have one folder per subject. So we're going to process that data right now in EEGLAB. So this assumes you've downloaded the data from Open Neuro. It probably takes about 30 minutes to download from Open Neuro. Then you open Matlab and start EEGLAB. If you don't have EEGLAB, you can Google EEGLAB download, and you'll get to the download link. You can download it. There's not much installation to do except to unzip the file and type EEGLAB on the command line, as we'll do now. So you're on Matlab, you type EEGLAB, and that's the graphic interface you end up with. And the first thing we'll do is BIDS tool, import dataset. So this is the dataset that downloaded from the web. And the BIDS tool, I show you the menu. You actually need the BIDS plugin to do that. That's not installed by default, so I'll show you how to install the plugins a little bit later. But this is how the data looks like. And so you have participants, and you have one file per subject. And so once we press this button, EEGLAB is going to import all the subjects, all the 4 gigabytes of raw data. And of course, it's not going to stay on disk. Also here, you see there's options when you import the data, because BIDS actually has some metadata associated with the files. So you have the raw EEG data, which is the time series. And then BIDS also have some channel location file, optional. And you also have some event file, which are optional. But you can decide to just get the data from the raw data. And sometimes the raw data also have events and channels information, or you can decide to overwrite the raw data, event information, and channel information with the BIDS meta data. There's also different ways to map the BIDS event files to the EEGLAB events. So this is a field to do that. We have a series of videos on BIDS that contain much more information about the structure of the file and how you do the mapping, etc. So here I'm just going to import the data. So now the data is imported. Of course, it takes much longer. The first thing, so if you remember the pipeline, is to remove the bad channels. Because what happens is that very often with your amplifiers, you have channels which recorded nothing, and you just want to remove them. So here you see these are all the EEG channels, and here I know that channel 61 from 64 contains nothing, so I'm just removing them. That's the first thing to do. Second thing is to clean the data with ASR. So here I'm just going to filter the data and remove bad channel. I'm not going to perform the the continuous data rejection, because this tends to remove the blinks, and then ICA cannot detect the blinks. Again, we have more detail on why you do all that, but here just filter the data, remove bad channels, removing bad channels based on the, if they're flat, I remove them. If they have too much line frequency noise, I remove them. Or the correlation, you remember the correlation matrix I showed you. If they're not correlated enough with other channels, they're removed. So we press OK. And again, this is going to take about 10 minutes. So it's not instantaneous. But here, this is a video, so it goes much faster. Then I re-reference the data. I like to compute IC on the re-reference data. You also have the options to interpolate the remove channel. So I remove channel with the clean raw data plugins. I can interpolate them before I do the average reference and then remove them again, which is more valid than just doing average reference with the missing channels. So computing average reference and then decomposed the data by ICA. Here I'm just selecting the default ICA. I have the options here to select PCA, so to reduce the data dimensionally by PCA, which is not usually recommended, but here we have removed one dimension when we did average reference, so we have to do that. And here I use PCA -1, which just tells ICA, okay, find as many components as the number of channel -1. And the reason I put PCA -1, here I have many data sets, so it's going to run ICA sequentially on every single data set. And so here again, I press OK, it's 10 minutes, then instantaneously. And then I use the IC label plugin. And the IC label plugin is going to assign a class to my components. I just use the default. And once I have assigned a class, I can set thresholds. So flag components. And here I put, okay, I want muscles. So I only want to flag muscles as artifact. I only want to flag ICA components as artifact if they have more than 90% probability of being muscle or 90% probability of being eye artifacts. So that's pretty stringent. But this way we only get a few components. I like to be conservative. Some people like to put loose threshold and remove lots of components. I don't think there is, as of today, a study that shows that one strategy is better than the other. So I keep up with my conservative strategy, which is to remove very little components. This is going to remove just 2 or 3 components per subject. So here it flagged the components, and then I removed the component from the data in that case. And the reason I do that is because I want to defer more artifact rejection. When you have a study like this, when you do group level analysis, you actually have the choice not to remove the components at all. And then when you do the group level, it removes them on the fly. Or you have the option to remove them from the data set themselves. And here I'm showing the second option, which is to remove them from the data set themselves, because I want to do further rejection of time windows. So I go back to clean ASR, and now I already remove channels. So I'm just using the last 2 parts right now. So the reason I did it this way, in this order, is that for this specific data set, there were lots of eye blinks. So if I was just checking all the options at the very beginning, it would remove tons of data with eye blinks. But ICA can correct the eye blinks, so I don't need to actually remove that data. So the way I do it is first I remove the channels, then I run ICA, remove the blinks and the muscle, and then remove the very bad portions of data before I process it further. It really depends on the data you're using. If you don't have a lot of eye blinks, you can just choose to run this clean raw data only once and check all the boxes. So it will remove the bad channels and remove the bad portions of data, after which you can run ICA to remove the remaining blinks. So yeah, it depends on the type of data you have. Here we found that for this data, this was optimal to use this strategy. So I select the last two portion, and again, This is the portion that use artifact subspace reconstruction, even though it doesn't use the reconstruction part, it just removes bad portions of data. So it's using the algorithm to detect the bad portions of data. And then you see the last checkbox here, additional removal of bad data period. This is just based on standard deviation. So sometimes the ASR algorithm misses some region, which can be captured by just looking at the raw data and seeing like it's very dramatic standard deviation in that specific window. If that's the case, just remove it. And you can also set the maximum out of bound channel. So for this specific criterion. So I press OK, and now I'm done cleaning the data. So I'm going to extract epochs. And if you remember, so there's 3 types of faces. And so here there was the famous first presentation, famous second presentation, famous third presentation, scramble, first presentation, second presentation, third presentation, and unfamiliar, etc. So here I have 9 types of epoch I want to extract. And again, once I do that at the group level, it's going to extract them for every single data set, one by one. So I select these epochs, and I want epoch -1 to 2 seconds and press OK. Then it's finally asking me whether I want to remove the baseline. In that case, I don't want to remove the baseline. So I'm done. I've pre-processed the data, and we're going to do that actually with a script, because EEGLAB, as you were doing that, wrote a script for you. So you can just save the script, and we're going to run that script on NSG. And so this is the script that's generated. The main advantage of using EEGLAB, like compared to other tools, is that we try to make things simple, in the sense like you run the same analysis in M&E, for example. This is like 2,000 lines of code, because you have to call all these detail functions, etc., to process all these subjects, about 1,000 to 2,000 lines of code. And there's actually a paper, M&E processing the same data, so you can see exactly what I'm talking about. And here we only have 100 lines of code, where we try to keep it as simple and as clean as possible. First, you import the data, so that's this line. Then you select the channels, you remove these 4 bad channels. Then we clean the data lightly. Then we reference, we run ICA, we run IC label, we label these artifacts, we subtract them. We clean the data more aggressively. We're using clean raw data. We extract the epoch and we create the study, in that case, the study design. And so what's here in the last part of this video, what we're going to try to do is to replicate what the author of this data set found for faces versus scramble. So they compared the ERP, event-related potential, for faces versus the ERP for scramble images. So they merged both the famous and the non-famous phase, and just look, what's the topography, what are the ERPs. So we're continuing, so we're moving forward. And so this is just a continuation of the previous video. And so here, now we move to the group level analysis. We're already doing group level analysis, but now we're going to do statistics at the group level. So you use this menu that's called study, and we're going to select the study design. And in this case, we want to compare famous versus scramble versus familiar. And I think we're actually going to compare the 3 types of images here. And so we're not going to group famous and unfamiliar. So we can see the ERP for each type of image. And so now we've created our study design. So that's 1 by 3. We're going to rename this. You can name different study designs. So this is stimuli. And then we're going to go and so we've done our study design. Then we're going to go and pre-compute ERPs. So that's pre-computing channel measures. And you can do that with channels. You can also do that with components. You will just do it with channel for simplicity. Here we can select a different time range for the baseline. So here -200 to 0, which was probably what they used in the paper. That's what we're using, the same thing. We compute ERP, and that ERP are being computed, and then we're going to plot the ERP measures, and we can select one channel. That's the one, the 65, that's the one they present in the paper. And you see the ERP for the famous, scrambled, and familiar. We're going to put them on top of each other just so we can see better the difference. And so here, this is how it looks. And we can compare that with the original paper. And you can see, well, we found something that's very similar to the original paper. We can also compute statistics. Here we'll do statistics on the scalp topographies. Interestingly, here we see a slow oscillation that they didn't mention in the original paper. So we have to do more analysis to see if this is real or not. But this is potentially interesting in this data, and they didn't mention it in the original article. Let's just look quickly at the scalp topography. And so here I'm selecting all the channels. I'm going to look at the 170, which is usually an ERP that's selective to faces. So the ERP between the scalp topography between 160 and 180. And so now I'm plotting the scalp topographies. So we saw the scalp topography, famous, scrambled, and unfamiliar. And I'm just going to quickly run some statistics using FDR correction. There's many correction for multiple comparison. Again, we have tons of video describing all of this in tutorial. And this is what we get in terms of topography. So this is an ANOVA across all the channels corrected for multiple comparison. This video, this presentation is actually a small part of a longer presentation, and it continues to do more advanced statistics using GLM and LIMO. So I invite you to check out the LIMO tutorial and the LIMO video if you're interested in doing more advanced statistics. But this is just an overview of, okay, we got some raw data, let's process the raw data. And here I jump through the parts that we're doing computation. So really, you really have to add 20 minutes to this if you really want to do the actual computation. But basically, we started from raw data. We imported the data, we cleaned the data, we ran ICA, removed bad components, calculated some ERP, and got some results in about 20 minutes on 19 subjects and 4 gigabytes of data. So it's fairly, and we got a script that we'll be able to run on NSG as well. And so the next thing I want to talk about is plugins. A few plugins in EEGLAB before I talk about NSG. So a few popular plugins. So we have the plugin manager. We'll use it to install plugins, the NSG plugin. And I'm going to show briefly 3 plugins. I don't know how I'm doing on time, Amit. And I think I'm like 40 minutes. I have like 30 more minutes.
Yes.
Okay. Because I want to leave some time for the demo. So out of the 130 plugins, I'm going to show you 3, which I picked because I worked on them and I think they're interesting. First, the general linear modeling EEGLAB LIMO plugin, which we developed in collaboration with Cyril Pernet. And this plugin allows you to do SPM-like analysis, so 2-level hierarchical general linear model on EEG data, except it's better. At least that's what Cyril claims, because he's using surrogate statistics for calculating significance. And so the way you do that is you have your design matrix as is SPM, and you have your first stimulus, which would be the famous image, for example. Second stimulus, which would be the scrambled image, for example. You can have a continuous variable, which can be the noise level in the image, and then you have a constant. You always have a constant in your design matrix, and then you apply this GLM on on one electrode every single trial at a given points, and you obtain some parameters, so beta parameter for each of these columns. And you do that through time, so you have your beta parameters varying through time, and you obtain some time varying beta parameters for categorical variables, so stimulus 1 and 2, and you can also calculate difference between condition at that level by subtracting the beta parameters and doing statistics on the beta parameters. You also have continuous variables, which are presented here, where it varies across trials. And then once you've done that, you can do a second level analysis. So again, that's very similar to what SPM is doing, where you've done that for every single participant and every single channels. And then you can do some group analysis on these beta parameter you obtain at every single level. And there's different types of optimization. You can use ordinary least square, you can use weighted least square. And in the lecture that's specific to this plugin, we discuss what's the advantage of doing that versus doing a mixed model where you do a GLM on all the subject at once and all the electrodes, etc. And basically the main advantage is computational. So we review all of this. This is another plugin here that was developed by Tim Mullen, and it's called SIFT, Source Information Flow Toolbox. And I'm just going to show you an example of how this plugin was used. This is a reaching task here where the subject sees a target and they're instructed to hold their movements. And then once the dot disappears at the center, start the movement and touch the target. So there's a period of retention of a few seconds in which they know the target, but they don't move yet. And this is what he obtained. So this is a complex plot, but here you can see different brain areas. So left motor area, right motor, left occipital, right occipital, ACC, material cingulate, left parietal, right parietal. So it has like 6 different brain areas. And then pairwise connectivity between these brain areas. And we see the pairwise connectivity in the time frequency domain. So on this axis, you have frequencies. On this axis, you have time. And so what's interesting here is the communication between occipital and ACC, in which you can see that during the retention period, you have this strong coupling between these areas in the direction of occipital versus ACC. So it's like perceptual area to frontal area, and then it disappears at the execution of movement. So this is the type of analysis you can do in EEGLAB SIFT. And of course, here I went super fast. So it's just the outcome of the analysis. Another one that's a new one that's also source connectivity. So it's very similar to SIFT. SIFT was using ICA components for performing the connectivity. This one used regions of interest, which are isolated based on fMRI Atlas. And this was developed by Stephen Hofer. And he had some code, and he sent it to me, and I put it in the plugin, and then he was really happy. We did that together. And so what this does is that this is the interface to the EEGLAB plugin, in which first you align your electrode with the scalp model. Then you align the atlas with your model, so you can do distributed source modeling, for example, using Loretta. And then once you have these distributed sources, you parcellate in different brain areas based on an atlas, and then you can calculate different measures, so pairwise measures, so time, reverse grandeur causality, or cross spectrum, or many others. And that's pairwise between all the areas. And the 3 you can use right now, you can use a volumetric atlas, so that's the full volume, or you can use surface atlases as well. And there's tools to align your head model or calculate your lead field matrix for this specific solution. And once you do that, you calculate the connectivity, and you can see, for example, which region are the most highly interactive. And you can also do the pairwise connectivity matrix between brain regions. So here we have 60 brain regions, or you have another representation like this one. where you see which area are connected with other areas. So this is more like a work in progress. It's actually not available in the EEGLAB plugin manager, but you can get it on GitHub. It is functional. So we're excited about using this new method, and also comparing with SIFT, because these are very different approaches. SIFT does partial directed coherence, so it considers all the area at once, and also it does indirect connection. This is just pairwise connection, so they can be spurious connection. And here you can use it with lots of brain areas. In SIFT, you're really limited to 9, because computationally and in terms of the data that's usually available, you're usually limited to doing 9 areas at most at the same time. So these are active areas of research. I mentioned several times our EEGLAB YouTube channel, in which we have lots of tutorial videos. We also have the EEGLAB tutorial with more like 300 web pages of step-by-step to analyze your data. And so this is the first part, and now I'm going to move to the NSG part. And so I also want to acknowledge all my collaborators and for EEGLAB, in particular, Young was a newer addition to our team, incredible developer. And then we have Ramon, who was also working on the core EEGLAB. Makoto contributed to clean raw data, which I showed you. Then we have Luca, who did the IC label, Scott McKeag, of course, which is the director of the center. So now let's move to the demo of using NSG for EEGLAB. And I still have a couple of slides, and so we're not going to do the demo right now. A lot of this work was done by Ramon, and so he offered the paper in NeuroImage on how to use this plugin. So you can look at that paper if you want more information. There's also a tutorial online on GitHub for using the plugin. So a lot of the information I mentioned here is available on the tutorial. So before I talk about NSG, I want to mention how NSG integrates into a whole infrastructure. We got a large grant from NIH to build a data archive for EEG data, which it's going to be coupled with NSG. And the way it's going to work is that, so bits data, you have bits formatted data that's submitted to OpenNeuro. And then we get a subset of that data because we cater specifically the bits dataset, which are high quality and that you can use. Unfortunately, if you look at all the datasets on OpenNeuro, lots of them are not usable because the data is not formatted right. It's still passed the bits validator, but somehow there's errors. So we calculate data metrics on all the data sets that go into OpenNeuro, all the EEG data sets, and we give more details about them. So that's the goal of NEMAR. And then these bits data sets will be available for processing on NSG directly. I'm going to process one here, but I'm going to load it manually. In the future, it's very likely that when you log onto NSG, you can actually select one of these either pre-processed data sets. So there's about 30 right now, 30 EEG pre-processed data set that we can make available. So you'll likely be able to select one of these in case you want to apply your analysis method to one of these data sets, and you'll be guaranteed that these data sets have all the information necessary to process them and can be imported in EEGLAB. And they'll be also pre-processed. So we will have a pre-processed version of these data sets. This is the list of data sets that we are planning to make available. So these are OpenNeuro datasets. They're identified by these numbers, and they have different types of metadata information. We also calculate the number of good channels, good data, and brain ICA components. So for each of them, we run them through a pipeline very similar to what I showed you, and we'll give statistics. So I'm going to show you three different ways to interact with NSG. First, the web portal. And I think Ted mentioned the web portal. That's also the way you use Neuron. And so we'll run a process on the web portal. Then we'll use the EEGLAB portal, which is called the NSG plugin for EEGLAB. And it's the same principle, except you're just on Matlab. So you don't need to go through the web and upload your file. Everything is done automatically to upload your file. And then the last part I'm going to show you is how to do it from the command line in Matlab. And for these, we have commands where you just give a folder and you say run on NSG, and it's usually as simple as that. And the plugin will convert to command line terminals. So you need to have curl installed on your system. If you have a Mac or Linux, it's usually installed by default. But for Windows, it's a little bit more, you have to jump for a bit more hoops to install curl. So you can only use this plugin if you have pre-installed curl on Windows. Again, for Mac and Linux, it's already pre-installed. And this interface with the NSG through the REST interface, and then you can retrieve back the results as XML, which are converted automatically for you. So, so what's the advantage of using NSG? So when you interface with NSG, you're using Comet, the Comet supercomputers. And so you're automatically allocated 24 cores and 128 gigabytes of memory. So you automatically have one node. And that's actually a lot. And most people interacting with a supercomputer have much less because they use different queues. This is a queue where you get a full node just for yourself. And so that's why sometimes it takes a little bit longer to get allocated compared to if you're using the computer on like the shared resource where you just ask for one core and much less memory. And this is right now, Comet, in a couple of months, and this is going to be to migrate it to Expanse, which has many more cores per node and also more memory. And you're likely not going to have 128 cores just for yourself. Probably you'll be capped at 64 cores, but that's still a lot. You have 64 cores. And here at Matlab has a big advantage because it's very easy to do multi-core analysis in Matlab. You just replace when, for example, you're looping for your subjects, you just replace your for loop by a par for loop. So the FOR you replace by par FOR, PAR, FOR, and it's automatically parallelized. It detects how many cores are available, and is going to, so here, for example, for this study, we had 19 subjects, so each core is going to process one subject automatically. And there is, when you transform your loop from a for loop to a par for loop, there's actually restriction on what you can do inside, but we made sure in EEGLAB that everything was compatible with par for loops, so you can parallelize using this method. And so here's just the comparison between laptop single core and NSG multiple core, and the time it takes to queue, estimate running the jobs, and then loading the results. So usually the gain in time is about 10, but we'll see what's the best use case for using NSG. So usually when you use a supercomputer, like before NSG, you need to have an account. You need to have time allocation on the supercomputer. And so that means writing an application to get time on the supercomputer, which is a lot of work. And also you need to be in a country which allows you to do that. So it allows you to write applications, etc. Not all countries open their supercomputer to all researchers. You need to have Unix command line knowledge, and you need to learn how to submit jobs to the supercomputer, which is not necessarily simple. You need to learn the queue system, and you also need to know the architecture of the supercomputer because usually you have different queues, etc. So NSG makes it simple because you only have to create an account and you press the button to submit the job. So that's the main advantage, but you get all of this automatically. And so also it's good to know what NSG is good for. And lots of people think, oh, the supercomputer is so much faster than my laptop or my workstation. even the one I just bought. And that's usually not the case. The cores are actually slightly slower, but if you buy a newer laptop or workstation, the core are slightly slower than your laptop, so they're not widely faster. They do have much larger memory, and we just saw, you know, the nodes, they have 128 or 256 gigabytes. So that's one main advantage of using supercomputers, is like if you have huge memory requirements, this might be the only solution for you, because you might not be able to afford a workstation with 256 gigabytes of memory. Also, it's usually slow. I mean, most laptops, modern laptops, require an SSD, so solid state drive. And then the supercomputer doesn't rely on SSD. They have huge disk, and these are old-fashioned disks, so the disk access are much slower. For EEG analysis, I mentioned the RAM, I mentioned the powerful loop, and the advantage that it's very easy to parallelize your job when you get assigned one node. And my rule of thumb is, like, if it takes more than one night to process on your laptop, it might be worth, you know, spending the time to try it on NSG. Or if you have huge memory requirement, very large data sets, like for example, you record whole nights with 128 gig channels, you can't possibly process that on your laptop. But you don't want to do that if it takes 4 hours on your laptop. It's not worth it. You just run it in the evening and get the result in the morning, which is what you would do on the supercomputer anyway. Nothing is instantaneous on the supercomputer. You run a job, wait a long time, and then come back and get the results. And so the process that benefits the most and for NSG are parameter search. So for example, you have an algorithm and you want to optimize a given parameters, where you're gonna have to run lots of jobs. So that's the perfect application for NSG, where you would either submit a long job that's going to take 48 hours to calculate, which is the limit. And by the way, you can't run a job that's longer than 48 hours. Or you're going to run multiple jobs, you know, which is each going to take an hour, but you're going to run 30 of these jobs, for example, one with each parameters. Or, you know, another use case is long processing applied to a single data set, so more than 8 hours on one data set. It's a good use case for using NSG. There'll be some frustration when you use, you know, supercomputer wait. You have to wait. It's not instantaneous. Also, you'll soon see that if you ask for too much memory, your job is automatically killed. So that happens often. You just require too much memory and your job just get killed. And so you wasted a couple of hours just submitting the job. And also debugging is not simple, because, you know, it's not like you're interacting directly with the Matlab command line. So I like to give another analogy here. So NSG is like, it's like you have a room of a standard workstation. So you still have to go to each workstation and start your process. So it's not fully automatic, and you still have to find some ways to aggregate your data once you start all these processes. But eventually, with effort, you imagine this array of computer, you can get where you want to be. And I also like to give this analogy that, you know, lots of people think that, okay, I have a supercomputer, it's like with the car comparison, it's like I had the standard Toyota, and now I have a Porsche or I have a Lamborghini, and that's not the case. You don't have a faster ZP car to do the job. What you have is a truck. You have a big truck, so it's harder to maneuver. It's bulkier, but you get the job done. And you can do things with your car you couldn't do before. So it doesn't go faster, but it can do much more that you could do with your car. So I prefer that analogy for, you know, especially for people who think, oh, it's supercomputer, just super fast. I also want to mention NSG for deep learning. One of the reason people want to use supercomputers is because we want to do deep learning. With NSG, So deep learning, it's a big black box. You throw stuff at it, you wait a lot and see what comes out. And usually what comes out is interesting. And deep learning usually runs on GPUs, and you cannot reserve GPU through NSG at this moment. Maybe it will change in the future. In practice, GPUs are about 10 to 20 times faster than CPUs. So you can still do deep learnings on CPUs. So for example, one SDSC, one node on NSG will be about 10 times slower than a standard GPU. The advantage of using CPU when you do deep learning is you can have much more RAM allocated compared to GPUs. But because it's slower and because of the limits, and you can't not run for more than 48 hours, it doesn't really make sense. You really want to run your deep learning on GPUs. So if you want to do deep learning, really the best alternative is there is supercomputers that offer a GPU interface, or you can also buy a GPU, which are very cheap right now. The gaming boards are actually very efficient, and they're as fast as the one you can find on supercomputers, except they usually don't have as much RAM. But that's a very good alternative if you want to do deep learning. I just wanted to mention that because I think lots of people, that's what they want to use supercomputer for. So let's move on to the demo. And here is our 2 slides I found about the gods of supercomputer. So let the god of supercomputer be with us, because sometimes there's weights, etc., unexpected things. So we'll hope everything will go smoothly. And I'm going to show you 3 things. First, I'm going to show you the web interface. So that's the NSG portal right here, and access NSG portal. So first, if you don't have an account, you have to register an account. I'm sure one of the questions would be, who can register an account? And anybody can register an account as long as you're non-profit. So you can be non-profit in anywhere, I guess even non-profit in China, and then you can register an account and get an account on NSG. And so there's a vetting process, and so it takes a few days. You register your account, then Amit checks your credentials, and then you get access. Then you press access NSG portal, so you get this login and password once you have access. And here, that's my interface, which is very similar to the interface you had for neurons. And the first thing you want is to upload some data. So here I already have uploaded some data, but I'll show you what I've uploaded. So this is here. So here I have downloaded the Open Neuro dataset, the one for EEGLAB. So if I go here, Open Neuro, I type EEGLAB. I go here, face processing for EEGLAB, download, and this is going to take like 2-3 hours to download this 4 GB from this website. But then I end up with this software, this folder right here. And for the purpose of this demo, what I do is I just keep one subject. So I remove all of these subjects, and then I have to edit the participant file to make sure there's only one subject left, first one. And so here my data is ready. I could process all the subjects, but it would take a long time. So if this has a chance of being done by the time we're talking. And then this is the pipeline. So here I'm going to start Matlab. I'm going to show you the pipeline. So that's the same script I showed you in my presentation with small changes. So here, drag and drop the script. So that's the script I showed you online. Load the data, import the data with bits, clean raw data, re-reference the data, run ICA, clean the data again, extract epochs, build the study. And here I'm plotting the ERP at the end. So we have some output. There's a bit of code at the beginning because there's some plugins here which are not available in NSG. So I just put them in the plugin folder. And for example, the bits import is not available in NSG. So what EEGLAB does right now, the new version of EEGLAB, will detect plugins if you have them in your folder and add them. In this case, I had to add a little bit of code because the EEGLAB version that's on NSG is an older version. We'll update it next week, probably. So we'll have a newer version and you won't have to do that. But right now, I had to add a little bit of code to add all these plugins. So this is just code that just scan plugins and add them to the path. So I move this script and these plugins to this folder. And I'm going to zip that up. Oops, duplicate. That's not what I wanted to do. I'm going to zip that up. And once it's zipped, it's about 500 megabytes. And once it's zipped, I will upload it to here, upload data, choose file, data, Matlab, test. Okay, I have my zip file. It's not done yet. It doesn't matter because I will not actually upload it physically. Okay, now it's done. So it's 500 megabytes. I select it here and then I press the save button and it will upload this. I've uploaded up to 5 gigabytes. 5 gigabytes on my laptop takes about 2 hours. But again, my bandwidth, it's just home. So it's not ideal. If you're at the university, it's probably faster. The bandwidth I've tested for NSG, when it gets your data, is about 3,000 megabytes per second. So you won't be limited by NSG. You will be limited by your home or university connection. So if I was downloading the whole data, it would take an hour. This would probably take about 5 to 10 minutes because it's just one subject. So I already did it. So it's here. It's the same. And so now I'm ready to run something. I'm ready to run that script, process DS that's in the folder that's together with the data. And so I'm going to create new task. I had run the same task 2 days ago just to check that it works. So I have it as backup. But usually when you arrive, you have no task here. It tells you how much CPU you're using right now. So I'm creating a new task. Open neural processing. Select input data. I'm selecting, I only have one file. Select tool. I'm selecting EEGLAB. And then set parameters. Here I'm going to say the script I want to run, and that's process DS. So that's the name of my Matlab file. That's the one I want to run. Here you see by default it runs for half an hour. Just to be conservative, I'll put 1 hour. And I'm done. I save the parameters. And so now I can save and run task. It tells me, okay, it's going to take some time, make sure everything works. So the script, you actually want to run it, simplify it and run it once so you're sure it works. And now it is queued right here. So it's queued on NSG. So here you see the status, it's added to the NSG queue, etc. And you can refresh it. And now it's staged for addition. In a few seconds, is going to be added to the actual computer queue. So there's the NSG queue, and it goes through the NSG queue and then gets added to the actual supercomputer. It's like a standard job on the supercomputer. And I don't know if you'll be able to see that. Even before it, even when it's still in the queue, it gets added. But it takes sometimes up to 1 minute to do that. It's not instantaneous. So let's assume the job is done. And we'll come back at the end, see if actually this job is done. So I run the same here. So this job is done. And so I can type view output. I can actually see the job here. You see, these are the next lines where it says, okay, this is the job on the supercomputer. This is it's loading results and it is completed. When it is completed, you also get an email telling you it's completed. So you know. And so let's view the output. And so we have the STD out. So that's the Matlab output. So it's create a Matlab session on the supercomputer and running a standard Matlab session. So if it crashes, you'll see where it crashes. You won't be able to debug it, but you'll see where it crashes. Then we have the error. These are files. This contains the list of zip files, so it's not very important. And this is the data. So once it's done, so the way NSG works is that it gets your zip file, it unzips the file, it runs the script, rezip it, and this is your output file. So it's not, you have to re-download all the data in addition to whatever result you might have. So here we're going to press download the results. So again, this is a 500 megabyte file. It goes much faster to download. Here it still says, 1 minute, but I have a copy, fortunately, in my download folder where I already downloaded it. So that's this one. So this is the one that's being downloaded here. And that's the one that's the same one I downloaded before. So I'm going to unzip it. And yeah, by the way, this other one is done. So you see it goes pretty fast, high bandwidth. So then you have this output folder that contains, again, the Matlab output. And then you have your original folder plus whatever file was saved in there. And you see this result file. This is the file I saved. And I saved the grand average, the ERP. So you can plot figures on NSG, even though there's no graphical output, you can plot figures. Here, it doesn't look too good because it's not, we didn't format it, which just was a proof of concept. But this is the ERP for this subject we process. So we pre-process the data exactly as I showed you, and then we get this final output. We could also have put all the subjects and get the same output I showed you online. But here we have a chance of seeing this process finished before we end. And so let's just check our task here. What's the status? We can see, okay, it is submitted as a job now. So it is a real job on the supercomputer. And we'll come back at the end, see if it's done. Now I'm going to show you the second way to do that. And that's the way through Matlab. So I have Matlab started here. I'm going to add EEGLAB to my path and start EEGLAB. I think I'm going to be done in less than 5-10 minutes, just for a minute.
Sounds good. Yeah, that will be good.
And so here,
we will not get kicked out, but I think we should be fine. Yeah, please continue.
Okay. So here, I have to install the NSG plugin first. So I go to the EEGLAB extension. So this is the way you install Limo, SIFT, etc. All these plugins. So there's 130 plugins here. And because there's so many, it's easier just to search. So you just search for NSG. Okay, NSG portal, 78 downloads so far. And I've just pressed install. So it's installing the NSG portal and it's restarting EEGLAB because it's going to add some menus to the interface. And then I go to tools, NSG tool, and here I have two menus, NSG account information, and then manage NSG jobs. So if it's the first time you arrive, you'll have to enter your NSG account information. Here, even if it's the first time I downloaded the file, I actually have the setting file with my information saved, and it found this file automatically because I had the plugin installed before. But if you arrive, it's going to be blank right here, so you have to fill in that information. This will be populated. This is the reference to EEGLAB, and this is the link to submit jobs, so you don't have to worry about that. But you'll have to enter your NSG username and your NSG password. Also, this is using a different system than the one that's online. So whatever we do here, we won't see in the other interface. It's going to submit jobs separately. So we won't see, we won't see the same jobs. It's using the same supercomputer, but different system. It's called the REST interface where we submit job through the command line. But it's the same accounts, so same login and same password. Here I'm going to cancel. And I'm going to manage NSG jobs. So I have a couple of jobs here I kept and saved just in case my jobs don't get finished on time during this presentation. So the first part here is the job list. And so you see I have two jobs. And that's the information about the jobs. They're both completed jobs. And so I can delete the job. So this way this disappears. I can see the Matlab output of these jobs as they're run. So this is the Matlab output that's downloaded from the supercomputer. If there's any error, the job will be in red. I can see the Matlab error, and I can download the job results in a very similar way that I would do online on the web interface. So that's the part that describes the job, and that's the part to submit a new job. So let's just submit a new job. And here I'm going to select a folder, and I'm going to select this folder, which is testing EEGLAB NSG. It's actually a folder you have in the NSG plugin. It's in the demo folder. Here I moved it somewhere else. And so this folder only had like 4 files. We're going to look at these 4 files quickly. And so we're here, data, Matlab, NSG, this one. So I have an EEGLAB data set, and I have 3 functions, which is run ICA functions. And here I see my 3 functions. And so the way these works is, let's open one of these. I'm going to drag and drop. We're going to run, they're all very similar. And they all start EEGLAB, load the data set, run ICA, plot the results. And I have the version that's called downscaled. This way I can run it here. I can test locally, make sure it doesn't crash. So that's what I'm going to do. So it's running it locally on my laptop, but it's a version that's much simpler. You only had 5 steps for ICA, so it did it right away. Okay, it seems to work. It's plotting the results. So now, as soon as this is done, I'll be able to submit it to NSG. And I'm going to submit the version that's not downscaled, the actual version. It's plotting 32 components here. Prefer to wait that it's done. Okay. So now we select the other function, which is the same, except it's not downscaled, and we run on NSG. And so as you run on NSG, you have to look here, it says busy. So it's busy doing something, it's busy submitting the job to NSG. And soon it's going to be submitted. So it's preparing the job, accessing NSG, submitting the job. And okay, now I have a new job. It's blue because it's still processing. I have this job already finished here, same job. I can show you the results. So this is the Matlab output. And so you can see it's just calculating ICA and then plotting the results. And then I can also look at the results. So I would download job results. So it's accessing NSG. Again, it's finding all the files. It's uncompressing the file right here. So I can go, it tells you where it uncompress the file. I can go right here and it uncompress the file in here, my current folder, and this one. So again, the output file is online, and that's the output of the job that was run on NSG. Simple. Here, I would never do that for an actual job. This is way too simple. It just takes a few minutes. I can refresh the job list here, see the status. So input staging, maybe it's already submitted. It is submitted, and that's the job number on the supercomputer. So the other thing I wanted to show you is that you can actually do something that's interesting in NSG, and that's called Relica. So that's bootstrap ICA. So I'm gonna, EEGLAB is right here. So I'm gonna load the data set. And so that's the tutorial data set of EEGLAB. Sample data, the one with data epochs. And then I have this Relica plugin installed. That's bootstrap ICA. So it's running ICA many times and seeing how components are reliable. So here I just run Relica. And so it's asking me what type of ICA do you want to use, a number of bootstraps you might want to use, the output, and this checkbox run on NSG. So it's going to submit the job automatically. You don't have to go through this other interface to do that. I'm not going to do it here because it takes time to package the data. It takes about 2 minutes to package the data and submit the job. But luckily, I have it done here, Radica. So I ran it before. So that's the output. That's the Matlab output of running Radica. And I'm going to download the job results. So it's accessing the job and it's downloading the results. And then I'm going to plot the results. So here it's interacting in real time with the supercomputer. So sometimes it hangs a little bit. So it just downloaded all the results. See, save them as Radica TMP. So now I'm going to go to Radica and load results from disk. And these are the Radica. See, Radica NSG. I go to Radica folder, Radica output, load Radica. So it's done loading and I can see the results. Basically, where I can plot, that's one of the standard Radica outputs. So you can see your cluster of components, or you can also use this one, cluster maps. And here, maybe we didn't load the right folders. Anyway, this worked before, so I'm not sure why it doesn't work right now. But it should work. Maybe I didn't load the right data set. So let's go back here, and then there's one last thing I want to show you. So this one is trying to transfer results. So it's still not done yet. So the last thing I want to show you is to run it, how to run it from the command line. And so for this, I'm going to here go back to this folder, and I'm going to see edit run multiple jobs. So that's this one. So see here, I'm going to use the same as the interface, but just from the command line. And I'm going to submit 10 jobs. And in this folder, I have a way to, so it's called multi. I have 10 functions to run ICA. They're actually identical. I could have different parameters here. They're just identical, but I just run it 10 times. And here I use NSG run to submit 10 jobs to NSG. So I'm going to do that. I'm going to copy this, copy and paste on the command line. So it just takes the folder, it gives a job ID and a file name to run. So every time it's the same folder. So you see one job was submitted, and it's going to take some time to submit them all. It takes about 1 minute to submit them all. And we'll see them appearing right here because we're using the same interface. We can refresh the job list here, and it would show them being added in real time. Once the job are done, and for the job to be done, you have to look at your email, or you have to look at this interface, whether they are now green or not. Then you can download the result of the job. So here I have the job reference. So I just download the result of the job. And here I have a simple command just to load the 10 ICA results. And then don't forget at the end to delete your job from the list, because then there's a lot of things in this list, and it just gets very crowded. So just refresh the job list here and make sure they're all listed. This one is done, by the way. So this is the one we started. You see it says completed. So I could look at the Matlab output. This is the same as the previous one, so I don't need to do that. Let's see, auto refresh. Oh, now it's refreshing automatically. So now we see, okay, I have 10 jobs submitted. They're all queued. So I can see what each individual job is doing. Submitted, submitted, submitted. So they all submitted. They already assigned a job ID. In all likelihood, within 5 minutes, they'll all be done. And I'll be able to run all of that. I'm just going to show you on the example as if I had actually done it. So here I would go to results, because I already run that code where it downloads all the results. Here it's updating the list. So So here I just downloaded the different ICA. So I have ICA 1, ICA 2, ICA 3. So it's just a proof of concept to show you what you can do. But here in 10 minutes, I can get 10 ICA decomposition. And then at the end, I delete the jobs once they're done. Before we end, let's just see if this job is done. And so view status of this one. And it says it's still submitted. So it's still waiting. So for some reasons, this one were executed faster than the other ones. And there's no reason, there's no way of predicting what's going to happen. Sometimes you wait 1 minute, sometimes you wait 30 minutes. So that's the end of my presentation. And yeah, if you have any questions, open for questions.
Arno, there are a couple of questions. Maybe I will read off from the question box. One question was pretty early, one of your slides, when you showed the two matrices, I think after doing ASR, good data versus bad data. So the question is, bad data versus good data would always look like the one shown in slides by Arno, regardless of any experimental conditions.
Yeah, it's going to look very similar. I mean, it's going to look like, there's some lines, and which this thing I showed you was for illustrative purposes. I don't think you can actually plot that unless you write a command yourself on the command line to calculate the pairwise correlation. So once you use the plugin, you actually don't see the correlation, but it would look very similar. It would look like some channels are not correlated at all with other channels, and we just label them for rejection.
I think that was the only question. There are a few other questions about the version and some other things that people from our side noticed. Those were covered in your talk, so they basically said they were covered in your talk. I think that is the only question.
Maybe just the one question on that. Does EEGLAB have channel location for bipolar montage data? Maybe just to say it for the audience.
So for bipolar montage, the channel location is independent of the referencing system. So if you have common reference, you have the same channels as if you have a bipolar montage. So you can have FPZ, FZ, etc. So they can be paired and be a bipolar montage, or they can be common reference. So EEGLAB has location for the standard channel names. It has template locations in different coordinate systems. So yes, we have channel locations. Once you have bipolar montage, you can also try to reference it to common average, which is much easier to process for practical purposes.
So Shubha, I think that answers all the question in the question answer box, right?
That's correct.
Right. So I think we will end now. We are just maybe 10, 15 minutes over, not much. So thank you everybody for attending, and we will make the video available after we do some editing of the text that it automatically encodes. And we hope to do similar workshops and tutorial in the future again. Thanks to Arno and Ted and Shubha for presenting. Thanks everybody, and safe wherever you are. Bye.